Welcome to the REU Virtual Symposium for 2023. This REU program was started under the auspices of Karen Holbrook at UGA and Provost Harris at Clark Atlanta University. We are now entering our 24th year of the program, long enough that some of our REU participants are now faculty at various universities. The program has been supported by the National Science Foundation under Sally O'Connor for 20 years. We're now in our 20th year of support of the program. And this program is also strongly supported by the Institute of Bioinformatics, the Department of Genetics, and the Department of Biochemistry and Molecular Biology here at the University of Georgia. I am glad to say that the support has also been nearly continuous from Franklin College of Arts and Sciences here at UGA, particularly thanks to the support of Dean Dorsey. Dean Dorsey is the Dean of Franklin College from 2012 to now, and has just finished his term as Dean of the college. And in a previous life at the University of Florida, he was also intimately involved in an REU program that he ran, and so he's very familiar with the impact of this on young scientists, and so it's absolutely appropriate that he actually introduce the REU participants today. And so I'm going to turn this over to Dean Dorsey, and I'll just mention one last thing, that this particular symposium is dedicated to Ernie, who provided the sandwiches for the REU participants all 23 years of the program. And please, Dean Dorsey, I'm glad to have you here this year.
Very good. Well, good morning, everyone, and thank you, Dr. Arnold. I wanted to first ask if my audio is okay this morning. If not, I suppose, how's my audio, Jonathan?
Okay, we're good. We're good.
Okay,
and I'm not hearing him.
Very good. Well, again, good morning, everyone. Just wanted to make a few remarks before you make your presentations today. So I wanted to thank Dr. Arnold for his leadership of this program over the last several years, and to welcome everybody. I know that it's been a lot of hard work over the past summer, but I hope your work over the summer has been both stimulating and fun. As Dr. Arnold mentioned, I'm a big fan of undergraduate research. My own experience goes way back to the summer of 1977, when I was able to participate in a summer research program for high school students in the Washington, D.C. area where I grew up. I had the opportunity to work at the National Bureau of Standards in Gaithersburg, Maryland. It's now known as the National Institutes for Standards and Technology, or NIST. And I worked on a project that involved precision polarimetry measurements of sucrose solutions. The details are a little fuzzy right now, but I do recall enjoying working in a lab alongside PhD chemists and physicists. My initial participation turned into an ongoing summer job. I was fortunate to work at MBS during every summer as a college student, working on a variety of projects alongside scientists and engineers. These experiences influenced my decision on a course of study in college, and I eventually majored in physics. I went on to earn a PhD in physics and became a faculty member at the University of Virginia and then at the University of Florida. While I was at UF, I decided it was time to pay back the benefits I earned from my undergraduate research experience, and in 1999, I co-founded a summer REU program in UF's physics department. We would bring 15 students to Gainesville every summer to conduct research in materials physics. I served as the research mentor for several of the students, and it's gratifying that some of them are now faculty members at colleges and universities around the country. I occasionally bump into them at professional meetings and conferences. Why is undergraduate research so important? It's the authentic experience of discovery, the pleasure of finding things out, to quote the irascible 20th century physicist and Nobel laureate Richard Feynman. It's about discovering and learning something that nobody else knew, or at least they didn't know it in the way that you know it. It's learning about new scientific techniques, working in teams, and alongside one of our talented UGA scientists. It's presenting the fruits of your work in accessible terms to your peers and to others. It's also about dealing with failure. Scientific research seldom proceeds in a linear fashion, and it's always fraught with pitfalls and failed experiments, dead ends, and misconceptions. Sometimes more things go wrong than right. And practicing scientists develop patience, persistence, and perseverance, which are important life skills that you can well apply outside of your scientific pursuits. And speaking of which, I know that many of you will be thinking about graduate programs soon, and as good students, I'm sure you'll have many options from which to choose. I hope you'll seriously consider UGA as one of your options. As I'm sure you've observed, we have outstanding faculty and great facilities. Please stay in touch with your research mentor as you consider your options. If offered a spot in one of our graduate programs, I hope you'll commit to the G. I hope your summer was productive and stimulating. I expect you have a better idea of the hard work involved in scientific research, and hopefully you've experienced the joy of discovery. I wish you all the best in your future academic endeavors. Thank you and have a great symposium.
Thank you very much, Dr. Dorsey. Your support means a lot to us, and I know it comes from the heart. So it's my pleasure to introduce our first speaker, Amol Abdul Jalil. She is from the University of Nevada at Reno, and she has two mentors, actually, Zachary Lewis in the Department of Microbiology, and also Abigail Devon. And she is going to be talking about a regulator in Neurospora crassa, which has ramifications for epigenetics. And so it's my pleasure to turn this over to Amel, and we'll hear about her summer's research.
All right. Hello, Dr. Arnold.
And
thank you
if
for
you'd
that.
load,
Let me go ahead and share my presentation. Okay. All right. Thank you for that. So let me figure this out. Okay, sorry. All right. So hello, everyone. My name is Amal Abdul Jalil, and this is my project that I've been working on this summer, modulating expression of the developmental regulator VSD1. So I just wanted to start off by introducing our model, Neurospora crassa. It is a non-pathogenic fungus. Neurospora is an already fairly well-studied organism, with it having its own molecularly validated gene knockout collection, and also in recent years having been fully sequenced. It is often used in lab settings to study circadian physiology and molecular biology due to it being able to grow very quickly and also containing haploid nuclei, which make it easier to identify new mutations and phenotypes. It is especially important to my research because it contains H3K27 trimethylated domains, but we'll discuss more about what that is and why it's important later on. So there are two ways that Neurospora can reproduce. In cases where Neurospora are in what is considered to be non-stressful environments with are my slides not loaded? Oh, sorry. There are two ways that Neurospora can reproduce in cases where Neurospora are in what is considered to be non-stressful environments with plenty of nitrogen to use as a carbon source. They reproduce asexually. So that looks like with mycelium forming specialized hyphae that will produce identical spores called conidia. So on the flip side of this, when Neurospora is experiencing nitrogen starvation, the stress triggers the sexual phase of the life cycle where we see mycelium forming protoparesthesia, which serves as the female parent. And when this is fertilized by a male parent, it forms a structure called paresthesia. This is where we see the ascus develop and form packs of 8 ascospores, which will then be ejected out of the paresthesia and germinate to form mycelia again. So knowing this and seeing these two different phases, we can kind of ask the question of like, what exactly regulates the transition from the vegetative to the sexual phases of the life cycle. So we can kind of look at this question by relating it back to what we do know about these kind of cell fate decisions using stem cells as an example. We know that stem cells have the ability to differentiate into any cell type. We also know that the identity of these cells are determined by transcription factors. And how chromatin is packaged. But something that we haven't that we have identified that has a similar function is the repressive protein complex polycomb repressive complex 2, which plays a heavy role in chromatin regulation and cell fate decisions. It is composed of 4 proteins, but the most important one or the most notable one for this research has been set 7, which is the catalytic subunit that is responsible for the trimethylation of lysine 27 of histone 3, which, or just a shortened version of saying it is H3K27 trimethylation. So this process is what causes the silencing of genes and is the domain of repressive chromatin due to the modified nucleosome being tightly condensed, which prevents transcription factors from binding with DNA. So with this in mind, we have found that cell fate decisions are actually determined similarly in fungal models like Neurospora. So in Neurospora, when the set7 protein is knocked out of PRC2 and the trimethylation of H3K27 can no longer occur, the target genes in this domain can no longer be repressed. So we have found that. So we can see the effects of this illustrated in a heat map, which, just to give a quick overview, shows the expression levels of RNA with each row showing a gene and each column being a condition, and the blue representing low to no levels of relative expression, and the orange showing high levels of expression. So in this heat map, it shows that in wild-type mycelium that none of the PRC2 target genes are relatively expressed, and mycelium with the set7 knockout have lower levels of expression. Lower levels of expression in genes targeted by PRC2. Something that we have found in connection to this also is that we see these structures called false parathesia, which are basically hypermelanized protoparathesia that don't form ascospores, forming when set7 is knocked out. So, in contrast to this, the levels of expression seen in parathesia, we can see are very highly upregulated in PRC2 target genes. So, in connection to this, we have it has been identified by my mentor that one of the PRC2 target genes, VSD1, may be the transcription factor related to the activation of the genes in these may be related to the activation of these genes in these repressed domains. She was able to find that when VSD1 is mutated, in Neurospora, that Neurospora loses the ability to make parathesia, which has led us to believe that VSD1 is necessary for sexual development to occur. So we can see this in this image, or in this chart here, that when VSD1 is mutated and attempted to be crossed as a female, it can no longer do. It can't do so. Okay. So that leads me to the focus of the Lewis Labs research. So they are interested in the, or they're basically interested in the establishment, maintenance, and spreading of PRC2 and H3K27 trimethylated domains. And so understanding this would basically help us understand the process, or give us insight to cell development and chromatin regulation. So the focus of my project is mainly on the sexual development of Neurospora and understanding the mechanisms behind what activates the genes necessary for the transition that we see from the vegetative to the sexual phases of the life cycle. I went about studying this in two different directions. In my first project, I looked into VSD1 and whether it is sufficient to induce the sexual life cycle in environments that would typically only facilitate vegetative growth. And in my second project, which I unfortunately won't be able to talk about just because of time, it focuses more on the cellular processes regulated by the set7 protein and how development is affected when it is knocked out of PRC2. So I did this by looking into, I looked at the conditions that were most favorable for false parathesia formation. And in a separate experiment, I basically looked at whether deleting set7 would affect a strain's ability to cross. So, so my goal for my first project is to overexpress VSD1 in Neurospora to see if this shifts it from developing vegetative tissue to forming parathesia. To test this, I assembled synthetic DNA controlled by a promoter to transform Neurospora. So I began by using PCR to extract VSD1 and the inducible promoters, TCU1, Q82, and VIVID, as well as the HIS3 gene, which will act as a guide for our DNA to integrate into the right spot on the genome. So these products were then taken and assembled to construct plasmids using the Gibson cloning method. And then from there, I transformed those plasmids using competent E. coli cells to basically generate multiple clones of the DNA. I then took the colonies, or picked the colonies that grew, and did a mini prep to isolate the plasmids, and sent it out for sequencing to see if they assembled correctly and if there were any mutations in the genome that we didn't want. So from there, I introduced those plasmids into Neurospora using electroporation, and I would have screened for transformants after that. So here are the results of the amplifications I did for VSD1 and the inducible promoters, their alongside with their resulting DNA concentrations. I also included flag, which is basically just a tag that we use to make sure that our DNA is within these cells. So after assembling all of the fragments with each of the inducible promoters, it came back that only the ones being led by QA2 were assembled correctly, which you can see illustrated in this image of one of the sample plasmids. I was able to use the plasmid to transform Neurospora, but I ran into an issue later when genotyping it. That where the image basically didn't show what we were looking for. So even though this may have been an indication that the transformation didn't work, I wasn't completely sure. So I decided to move on and induce the promoter using quinic acid anyway, just to see what would happen. So the transformants ended up not having the phenotype we were looking for, and only growing normally, growing conidia. So there were no visible paresthesia. But since the promoter QA2 is normally weakly expressed, I thought that that might have been the reason why we saw this. So we decided to do a Western blot just to see if that could give us any indication it did work. But unfortunately, the results confirmed what we originally thought, so it didn't end up working. So the next steps for this project would be to retransform QA2 and vivid plasmids into Neurospora, and also TCU1 after reassembling it. It is essentially, this is essentially done just to compare results and ensure that we see it do what we see is due to the overexpression of VSD1 and not the inducible promoter. And then from there, we can induce the transformants and screen for the formation of vegetative or sexual structures and back cross them. So once we've done all this, we hope to see overexpression results similar to this chart, which shows that when VSD1 is led by a very strong promoter and inserted into Neurospora, it can form what looks like paresthesial cells. And I think I accidentally uploaded the wrong presentation, so I didn't get to have my picture here for my acknowledgements, but I just wanted to say thank you to the Lewis Lab for allowing me to have this amazing experience. And I've learned a lot. So it's been great. Thank you.
Thank you very much. This is wonderful, and the audience can just type in any questions, and I will read them back, and I will respond. So go to your screen and type in questions, and while you do that, I will just take a liberty to ask a few questions myself. So does the VSD1 gene have homologs in other organisms that, yeah, that's my first question.
Well, it is, it's in a lot of, I'm not sure about VSD1, but I know that PRC2 it is in a lot of different organisms, in a lot of higher eukaryotes, but I'm not sure about VSD1.
Okay, but SET7 is the polycomb gene, and it's in things like, it was first probably discovered in Drosophila.
Yeah,
Is
that's what I think from what I've read.
Yeah.
Yeah.
And one of the interesting things about polycomb genes in Drosophila is that they have effects at multiple life stages. So
yeah.
So does that kind of happen in, you were talking initially about looking at some of the other effects of SET7.
Yeah. So when SET7 is knocked out, we basically saw like these structures that looked like parathecia but weren't actually parathecia forming, which was kind of interesting. So they were basically imitations, but they weren't like the actual thing. So that's kind of there's a connection there, kind of.
Right. Are there sort of functional analogs of ultrabithorax or trithorax, these other positive regulators that work with the polycomb genes? Are there things that provide sort of positive control over the chromatin area? I know this is acting as a repressor.
I'm not sure. I'm sorry.
Okay.
Yeah, no. Most of I haven't really gotten the chance to read about that or learn about that yet, but yeah.
Okay. And I'm just waiting here for people to load questions. I'll ask a question about your methodology. The way that you put in the knockouts, was that by CRISPR-Cas? Was it, you said it was targeted, or did?
What do you mean by that? I'm sorry. Can you repeat your question?
Yeah. When you targeted, you did your transformation, was the transformation assisted with CRISPR-Cas9?
No. So we basically just used the His3 gene, like when we made our plasmid, to act as a guide for it to integrate into the genome. But at least to my knowledge, I don't think we used Cas9. We did not. We didn't do that.
So did the plasmid have two regions within the His3 gene on the plasmid, or simply one region?
So sorry. I think it was in my presentation, but or at least like the little chart that I showed. It kind of showed it was kind of cut off, but I believe it's only one, but I might be wrong about that. I might be wrong about that. Yeah. I just noticed like halfway that I
Right.
might have uploaded my not fully
Right.
completed presentation here.
Okay. Okay. So I'm going to
But yeah.
remind everybody throughout this symposium, you can submit questions. I think they're at the bottom of your screen. As you listen, you can do the submission. But I will just finish. I think we have to stop here. I was gonna. It's 9:30, and I want to thank you for this wonderful talk on what has got to be one of my favorite organisms. So it's real close to my heart, and thank you for the wonderful presentation, Amal. Okay.
Thank you.
Okay. So in the same spirit of Amal, we're still going to stay a little while on Neurospora crassa. It's my pleasure to introduce Esther Hassan. She's from Belmont University, just north of us in South Carolina, I believe. And her mentor was Dr. Jia-Wei Chung, and she is going to tell us one of the main stories that's come out of Neurospora, which is understanding our own ability to tell time through the biological clock. And what she's going to do is tell us about work to characterize the biological clock and how it works in fungal filaments. And so it's my pleasure to introduce Esther. And again, you can go to the bottom of your screen at the end of her talk and type in questions, and I hope you will. And as you get familiar with the setup, and please proceed, Esther. Thank you.
Thank you, Dr. Arnold, for that introduction. Make sure you can see my slides. Okay, great. So as you mentioned, I will be talking about Neurospora crassa, and my research is focusing on measuring the infection zone and how that relates to the synchronization of these single filaments. And I'll explain what all this means later on. Before I begin my presentation, I just want to give a little outline of what I'll be going through. Sorry. So first, I'll give a little background behind my research, and then my motivation with research, my hypothesis, my aims, and the experimental design procedure that I went through. And lastly, I will show you guys my results. So starting off with the background, as you mentioned before, I am looking at the area dealing with biological clocks. And these clocks are essentially our natural timing devices that coordinate our circadian rhythms. And circadian rhythms essentially are, in all organisms, they control our mental and physical behavioral changes throughout the day and the night. And these clocks, they enable the coordination and execution of complex cellular processes. And this involves developmental programs, intracellular signaling, and etc. And as mentioned before, I am looking at Neurospora crassa, and this is a bread mold and is basically a model organism used to study these clocks. So dealing with Neurospora crassa, it is challenging to measure their growth because of their extensive cellular branching, as you see here. So to combat this, we use a microfluidic system to be able to trap a single filament in a channel, and we can study that filament over a prolonged period of time. And microfluidics is a relatively new physical study, and essentially is a system that manipulates small amounts of fluids in small channels. And this is a great way to be able to study things at a micro scale and have a more controlled environment. So going on with motivation, we know that synchronization of cells is important to know because it helps us to be able to study specific events throughout the cell cycle and understand more on the different things that happen in these cell cycles. So essentially, we're trying to find out how these single cells are able to synchronize with each other. And we know that in filamentous fungi, nuclei are transported over long distances within their hyphae. And one way of doing this is looking at the advection zone. And so advection is the movement of solutes in a certain area with the flow of the liquid. So in my case, I'll be looking at the nuclei moving with the flow of the media that we are putting in. And so my hypothesis is basically that these single cells of Neuris procrasta, that they're able to synchronize with each other under the influence of the advection zone. And so the advection zone is essentially the area I will show later in the graph where there is one area of high velocity and then a drop, extreme drop in velocities, and the rest of the velocities further on are moving at a more relatively slow rate. So that is what we are calling the advection zone. So the aims that I wanted to achieve with this research is, number one, I needed to calculate the nuclear drift velocity, as mentioned before, by obtaining their displacements and their time and their speeds. And then I wanted to determine the size of the advection zone as a result, again, in the synchronization in filaments. And also I wanted to see if there was a daily dependence of the size of the advection zone, whether the size be larger in the beginning and gradually get smaller, or vice versa. And so mentioned before, we are using a microfluidic system. And so here is an example. As you see here, we have our device with an inlet where we put media and an outlet to where we get excess media. And we have two plugs to keep the pressure maintained within the device. We also have a hole in the middle to inject our agar plug with the fungi, and it will be able to grow within these channels. And as you see here, this is what we call serpentine channels, and the filaments basically are trapped within these channels, and we're able to look at them individually. And this is right here, the 2D hypha clock model that was created before I came. And we're essentially trying to find the size of advection zone, which is around this area right here, and hopefully get a more accurate number. So now we're going to go into my actual experiment and procedure. And I basically have two parts for this. The first part is the actual experiment part. And first, I created the device, as you saw before, a 64 millimeter device with the use of a syringe and tubing. And then after we inject our agar plug and the filaments, we allow the filaments to grow overnight, and we begin to check in on them daily the next day. And so as you see here, the microfluidic device is placed under the microscope, and we use it to take images of both bright field to see the actual physical filament, to see the different characteristics and what that could add to the velocities of the nuclei here in the fluorescence images. The next part is the image processing. So after I take all the bright field and fluorescent images of certain channels, I then take the fluorescent images and I place it into a program called Cell Profiler to be able to track the nuclei. And by doing this, I take a crop section in the middle of each segment of the filament, and I try to maintain the same size to be as accurate and precise as possible. And I take these cropped images and track it, and then I place those images that Cell Profiler produces, and I put it into another program called ImageJ. And essentially it takes the individual images that Cell Profiler produces and puts it into one image, as you see here. And it basically helps me to make sure that my tracking is done correctly. So going off with the imaging, a way that I categorize my data is basically breaking off different sections of the channel that I'm looking at. And for example, this is section 1. And so for each section that I take, I make sure to have 6 segments in each section, just to make sure I'm more everything's more organized and everything's the same. Certain sections will have, certain channels may have more sections than others. So I just make sure that I can keep track of how many sections each channel have. So now we can look at our results. So for my experiment, I ran it 3 times and I got their velocities, plotted it in MATLAB, and I essentially took all the velocities and I plotted it on a speed versus distance from a way tip plot that you'll see later. I then, looking at these plots, I did an eyeball estimate of where the vector zone would be, and then I created a range. And this, for example, is from experiment 2, and this is essentially the different size of the infection zone here on the different days. So looking at this, for example, for experiment 2, you see there is some increase along the days, which was interesting, but some of them didn't really move, as you see here in this channel. So we decided to look further into this. Just a quick note here, and this is an example of a distance versus speed plot that I was talking about. So we were trying to look for this initial high speed, as you see here, and then a gradual drop or deep drop of velocities, and then velocities then leveling out as you go along away from the tip. And again, you see here on day 3, for some plots, they didn't look like that. It was more having an increased speed from sections away from the tip, as you see here in day 4 and 5. And this pattern was seen a lot more with experiment 1, which was the one I was practicing and testing on. So we decided to use data more from experiment 2 and 3. So then this, for example, is from experiment 2. We took all the sections and channels, velocities and their distances, and we plotted it all on one plot to see where the general infection zone would be. And so eyeballing it, we decided to say that it was around 800 microns. So then we split it into 2 groups. We have our first group here, which is before 800 microns, and we labeled it 0. And then everything above that, we labeled it 1. There are some outliers here out away from the tip, but we do see the initial drop here in the beginning, so we decided to focus on that. So then with those 2 groups, we decided to run a Kruskal-Wallis test with assuming the size is 800 microns, and we got a p-value of 0.0279, which indicates that the infection zone could possibly be around 800 microns. We also ran this test with the size that they got before I came, which was 672 microns, and there was a significant more value, 0.0052. So knowing this, I ran a Friedman test to see if there was a daily dependence using this 672 micron size, and I used experiment 2 and 3, and I got a p-value of 1, which suggests that there is no significant dependence on day. This could be due to me not getting all experiment 3 data. Unfortunately, I think I had like 2 days left, or it might be some other factor. But based on the data that I did use, there was no significant dependent on day. So for further studies, we'll determine to see if there is another factor that could create increased speed in earlier sections. Also to check the significance of each group, the 0 and 1 individually with the Kruskal-Wallis and Friedman test to see if there's a difference, and also to be able to run any experiments changing the flow rate of media that we were putting in to see if that will also change the size of the infection zone. So here are my references, and as all of you guys today, I just want to give a quick thanks to Dr. Arnold and Dr. Gia for helping me along the way and learning all these new programs and working with the microscope and devices. Thank you guys so much. But it was an amazing experience that I went through this summer. And that is all I have.
Thank you. Thank you very much. That was a wonderful presentation. And so I was curious from the, you introduced a model which sort of makes predictions about the size of the advection zone over day. And what was the prediction? Would the model predict the size of the advection zone to vary with day or not? That's my sort of first question.
Yeah, I think that's what we we're expecting based on the model that the size or the day would have some sort of significance with the size of the infection zone. And further, like, as you know, we were doing these tests and it was looking like it did have dependence, but when crossing with experiment 2 and 3, it didn't seem to have a dependence with the Friedman test. So I don't know if there's another variable that I'm not looking at, or maybe number of channels. I don't know, but
but the when you look at added the additional days of data, it was probably from days 1 through 3 or 4, something like that you used. And you have data up to like day 6, right?
Yeah.
That we still have to code. Okay. So it's still, from the data that you've looked at so far, there doesn't seem to be a dependence on day, which I think is the prediction of the model. And then what would also be interesting is to understand whether or not there are any genes in Neurospora that actually affect the size of the advection zone. So you have any thoughts on that?
I don't know for certain if there are any genes that relate to the size of the infection zone, but I do think that would be something interesting to look at.
Okay. There is a question, somebody from the audience, and I'm just trying to find it here. Here it is. This is anonymous, so great talk, Esther. When you are looking at the movement of nuclei in the channels, do you account for the different direction they're moving? That's the question.
Yes. So no, there are negative and positive, like based on the direction of the movement. So that's why I decided to use the speed, because it is a scalar unit and not the velocities, just to make sure everything is like easy to use. Yeah.
Okay, so you're looking at the speed, which doesn't have a sign with it, but some of the complications of measuring the speed. Can you tell us some of the challenges of measuring the velocity of the nuclei that you had?
Yeah. So sometimes when measuring the velocities, Cell Profiler might not be able to track it well, either if it's moving too fast or there's not a lot of nuclei for it to be able to sense it. And that caused some issues that I had to deal with. Also changing the threshold of the actual in the program to make sure that I can get as many nuclei to create. That was kind of tedious.
Those are some of the challenges.
Yeah.
So I'm wonderful talk, and I think we have to move on to our next speaker. And wonderful job, and we'll and I'll hopefully see you this afternoon before you guys take off. So we'll be in the lab to congratulate you. Okay, our next speaker is Antonio Holmes. He's from University of Maryland, Baltimore County. His mentor was Dr. Xiaoying Zhao, and he is going to look at a mutant that causes cancer in Cocker Spaniels. And so this will be of interest to everyone who has a pet dog like I've had in the past, and I've even had a Cocker Spaniel. So it's my pleasure to turn it over to Antonio Holmes.
Well, as long as y'all can see my presentation. Okay, perfect. Thank you for the introduction, Dr. Ono, and my presentation will be about the increased susceptibility of adrenocarcinoma and Cocker Spaniels with a DLA88 gene mutation. Starting off with, I will go over 5 sections, starting off with background and significance. So the project originally stemmed from the recent work of a bioinformatics graduate in Dr. Giles' lab. He created a software that hypothesizes where mutations will be from the data that you input. It was observed that cocker spaniels were likely to have a tumor that originates in glands, and tumors that originate in glands are called adenocarcinomas. Connecting research also confirmed that the survival rate for 1 year is decreased down at 59%, 3 years, 40%, 5 years, 13%. So this is a problem now. And then according to Cancer Stats 2023, for humans, there will be 820 new cancer cases. Cancer deaths as there was in 1,958,000 new cases in the United States. So, introduction. Starting with so when we originally started, the problem statement was cocker spaniels are more prone to adenocarcinoma, decreasing their life expectancy. Research question was, why are cocker spaniels more prone to cancer than most other dogs? Rationale is that there will be a mutation in the cocker spaniel genetics that makes it more susceptible to adenocarcinoma. Hypothesis, looking at the software, cocker spaniels with DLA 88004002 allele has an increased susceptibility of cancer. The objective of our research is to identify what makes the cocker spaniel more susceptible to adenocarcinomas to the development of novel treatments. After we confirm this objective, next objective will be to transfer the study into humane studies, the results into humane studies. Start with our descriptions. So, okay, they popped up already. So, DLA88 immune response plays a critical role in recognizing foreign substances in separating from our own body cells. So, DLA8 is a MHC gene, and once again, DLA is the system of genes involved in immunofunction. 88 is the specific gene locus. 004 specific allele variation. Some people call that the level 1 identification. And then 02 helps identify the particular allele variety. And some people call that level 2 identification. So next we be talking, and the first problem that occurred is that the forward primer cannot distinguish the forward primer cannot distinguish DLA88 from DLA64, but the issue was resolved by development of primer that bonds to the DLA88 and not to a reverse primer that bonds to DLA88 and not DLA64. Next, we got nanograms per microliters, and that's just the concentration measurement, so the amount of DNA you have in 1 microliter, and the absorption rate 260 and 280. The rates of absorption just assesses the purity of the DNA and RNA, and these are both the results of a nano drop. And the acceptable purification for DNA is 1.8, which means if it's above, most likely your sample is contaminated. And also, if the sample is below 1.8, you most likely have a protein or another contamination that is more highly sensitive to the UV light. And then after that comes steps in between it, which is double digestion and transformation. But the final step before we move on to our next objectives will be sequencing sequences, and it's just the process of DNA replication and sensitivity to detect mutation, distinguish between inserted or deleted. Methodology. So this we'll be going over 2 kits, and that's just the quick DNA, the quick DNA mini prep kit, and the second, and multiple other kits that we won't touch on right now, but includes double digestion, ligation, transformation, and more. And the highlight of this would be the procedure. Yes, the highlight of this is the procedure where the errors that came most during my project, and that's doing purification of the 3 whole blood samples using the kit, measuring the sample on the nano drop, modifying it, and repeating these steps to get better results. So this is just the overall outline of standard procedures. I'll let you read, but I will talk about it in bigger view. So the purpose is just to purify the sample so we can send it off for sequencing. But before we send it off to sequencing, we have to leads to transformation. Transformation is just when you feed it to E. coli. It's, in simple terms, you feed it to E. coli and you send it off. So the change that we made was including emptying the collection tube and distinguishing on the last spin, so that would save money at the end. But the major steps that we changed was additional drying steps where we started to see results became better, distinguishing the flow as we go. And instead of using buffer, we use water. So results. During our first one, you can see I highlighted the two sections. The nail drops per microliter was doing pretty well or overall in the beginning. Next came 4.4, which was bad at the time. Also for the 260, we was getting terrible results. Remember, we said for purification, we're looking forward to 1.8. Remember, we was getting 9.1, 2.2, and so we had a lot of outliers and no standard for the results. But after our modifications, we see the results started grouping up better, except for one outlier that was tumor sample that we ran with blood that returned back with absorption rate 5.44, which is an outlier compared to the other ones. But our other results are very outstanding compared to the previous. So closing remarks. So our next objective is just to replicate good results with the modified procedures. Attempt to use the modified procedures with cocker blood against send cocker blood off as gene. For sequencing, identify the mutation. If not, then where's the mutation, and identify if the mutation matches other sequence samples. So these are my reference and my acknowledgments. Thank you to Dr. Giles' lab for helping me and supporting me through this process. And thank you. Any questions?
Thank you very much for that very interesting talk. And did you, do you have any hypotheses about what other cancers this particular mutation might cause? You're focusing on, the one particular cancer. Yeah, but you mentioned that Cocker Spaniels have problems with an array of cancers, right?
Yes.
Yeah.
So adenocarcinoma is the umbrella, and the various cancers that come under it would be anything that includes a gland. So that's such as breast cancer, prostate cancer.
Mm-hmm. So all of the, okay. So that, and and I'm just going to remind people, you just go to the bottom of your screen and you can submit questions. I'll let you try that too for Antonio. So the other thing I was curious about, when do you get the fruits of your labor? Does the sequence, are you going to play with the sequence once it comes back from
So once it's sent out, normally the results come back the next day, and that's when the bioinformatics students use it and identify the mutation or genotype.
Okay. So hopefully you'll get to play with that too some. So yeah. So what is unique about the dogs as far as telling us something about cancers in humans?
So as I was talking about, the broader impacts would be from transitional studies. So making a similar procedures with modifications that can be used on HLA, which is just human leukocytes antigens. And a great example of that would be HLA-G expression, which is associated with poor clinical outcomes in patients. So that's the next leading point.
So the goal is to get some kind of immunotherapy up and running in the dogs with hopes of translating that into human systems.
Yes.
So, okay. Okay, I think we're basically right on schedule. And what we'll do now is thank Antonio. I haven't seen any questions, but don't be shy, audience. Just look at the bottom and put the questions in, and I'll read them back. And thank you very much, Antonio. And so now we'll have a 15-minute break, and I'll see you in 15 minutes. So don't go away. Okay. Welcome back to the 2023 REU Symposium, kindly brought to you with the support of the National Science Foundation and the Department of Energy. It's my pleasure to introduce the next speaker. He is part of a large DOE project under the leadership of Jeff Bennetson. And the data that he'll be talking about was collected from the laboratory of Nancy Johnson at Northern Arizona University with the support of Sedona Span. It's my pleasure to introduce Stephen Easton. We'll be looking at this data. I'm actually the mentor on this project, and he will be looking at the relationship of the colonization of sorghum by arbuscular mycorrhizal fungi and the developmental stage of the host plant. And it's my pleasure to introduce Steve. So please go ahead.
Thank you, Dr. Arnold. So for my research, I wanted to look into if there is a correlation between AMF colonization and the actual developmental stage of the sorghum bicolor plant. So first, a little background. What is AMF in the first place? It stands for arbuscular mycorrhizal fungi. They are soil microbes that colonize the majority of plant roots, and they end up forming a symbiotic relationship where in exchange for carbon, they provide the plant with additional nutrients such as phosphorus and nitrogen. This image here is a slide image with the AMF stain purple. So next, for developmental stage, the way I went about doing this, so I wanted to find a good standardized method in order to break down where each plant was. So I found this paper, and it helped me out with saying each stage is either at a 1, 2, or 3, with the first stage being characterized by vegetative growth, such as the image at the bottom left, where it is just starting to come out of the ground and starting to have leaves and tillers. Growth stage 2 is roughly in the right side image, the middle 2 plants before they start flowering. It's when they start making reproductive structures such as the panicle, and it starts making the maximum number of seeds that are made for its lifetime. And then the final stage will be represented on the bottom right image by the 4 plants on the right. Is the final stage where it is the grain filling, and it has begun flowering and continues to do so until essentially it finally dies. So what are my goals here? Obviously I was curious if there is a correlation between the AMF colonization and then the developmental stage of the plant, but I was also curious, would it become more parasitic or mutualistic as the time went on, the symbiotic relationship? And so for this case, parasitic meaning that as the AMF is not giving as much as it's taking, and mutualistic, they're both having an even exchange and both happy and healthy. So my overall hypothesis was that knowing that AMF is known to be able to manipulate signals and processes of the plant so that their relationship can take place, such allowing it to uptake more nitrogen and phosphorus and it to take the carbon. Knowing this, I figured that as the plant grows and develops, it's going to release certain signals and processes for those developmental stages, and that should have an effect on the AMF colonization over time and the overall plant's health due to possible interference or otherwise overlap. So how do I want to go about doing this? My initial procedure, I wanted to collect data from an Arizona time point study that was conducted, compile and convert those values into colonization percentages for each time point that was taken. And then that was going to be done using the McGonagall line intersect method. That method is a method to estimate colonization based upon the intersections between the microscope eyepiece crosshair and the actual roots themselves at a rough magnification of about 200. And then using each time point in the colonization percentages, I want to perform analysis to verify that there was indeed a correlation. So very early on, I had a little issue. When I collected the data, there were supposed to be 6 time points with a mature final, quote unquote, 7th time point with varying weeks. But as you can see here, Time points 1 and 2 and 3 were scored, and the mature were scored, but due to the poor quality of the roots for 4, 5, and 6, they weren't able to be scored. And when I looked into the data and looked at time point 3, they were actually, as it states there, very generally poor, so I couldn't really use any of the data in time point 3. This was a major confounding limitation. So the main thing I could use was time points 1, 2, and mature. So going forward with those, I utilized those with mature being labeled as time point 3 for many of my analyses. So first, I want to look at the percent colonization for each accession and plant, with the PI numbers being on the bottom here, and the percent total, or P-tote, of colonization being the y-axis. And as I initially saw, there was wide variation among most of them, except for a couple that had either none, as you can see on one accession, but most of them were very varied. The first thing I noticed, though, was the confidence diamonds were very skewed and very large, which was concerning. But to confound this, I moved forward and began a look at the colonization to see if there was a time point difference. And as you can see here, the time points 2 and 3, 3 being mature, were both very similar in the way they were broken up. But time point 1 is very odd. The reason for this was because when they were taking the time point study for time point 1, since the roots were so kind of fresh and they weren't that developed at that time point, they had a hard time using the McGonagall line point method in order to score them. So they used that as a cheat sheet and came up with a variation of it using a scale of 1 to 5. They said if it had a certain structure, such as a vesicle or an arbuscule, they said 1. And if it didn't, they said 0. And so this led to a scale of 1 to 5, 5 being the most colonized, 1 being the least. And so when they had a separate conversion table in order to turn that into percent colonization. And because of that, as you can see, it led to a bit larger skew for time point 1 than time point 2 and 3, which is more where the limitation became apparent going forward. But I wanted to make sure, so I did an ANOVA table analysis, which statistically showed there was variation across the time points. Now, this ANOVA table here, as some of you are probably already noticing, says no, it's 0.4489, which is statistically insignificant. This table came back when I did an analysis on the what is called, I labeled as AP-tote values in the time point, which is also the chart to the right. AP-tote being I took the percentage colonization values, and then I turned those, use a triangular conversion to try and make the data a little more uniform to get rid of the effect of supreme outliers. But as you can see, it came back showing that there was insignificant. But just to verify, I tested the original percent colonization values, and as that came back on the ANOVA table on the top here, that showed that there was significance. So because of the two different, one saying significant, one saying not, I wanted to do further confirmation, doing a Freeman's ANOVA table, and that did confirm with a 0.011 value that there was significance. So going further, I wanted to see where was the differences at. Was it across all time points evenly, or was there specifics? So doing the final Freeman's ANOVA on the bottom, it was a multi-comparison, and that showed that there was significance. The biggest significance, though, was between time point 1 and 2. Time points 2 and 3 were very similar. So with that, I realized that time point 1's scoring method might have led to a vast skew and showing significance where there could not be. So this was the chart that it came out with, which the time points are on the left, and the mean ranks, where it does a ranking of the whole breakdown, and as the mean, then it shows the means up here. Well, as you can see, time point 1 is the highest and way towards the right compared to time points 2 and 3, which are both more either centered or to the left, and they were very similar, while time point 1 was vastly different. So having this showed me that I couldn't really, with any level of confidence, state that there was or was not correlation due to the difference in scoring method. I couldn't say with confidence, oh, this only affected it slightly, or this was the reason there was significant difference. So I said, okay, what are ways to maybe go forward with this, or what else could be done? So a future study into this could involve doing a new study. It'd be very intrusive and time consuming, but it would be worth it with regards to keeping a single scoring method this time for all the time points, even if it doesn't work perfectly. That makes sure everything, you don't have a massive data skew like I encountered, but everything stays relatively uniform. Investigate some of the other outside factors that I was not able to, that may have changed the results, such as disease or weather. Was there a drought? Was there a massive disease that hit all the plants, and that could lead to plant health decay? So that would affect whether or not you could say the developmental stage is affected. And then finally, looking at, was it parasitic or mutualistic over time? Is the colonization levels high and the plant health bad, suggesting parasiticism, versus both the colonization levels are high and the plant is very healthy, suggesting a mutualistic relationship as it went forward. These are my lovely references. Thank you for listening to me today. Do you have any questions?
Again, the audience can submit questions using the spot below at the bottom of the screen, and I'll just read the questions to Stephen. So while you're typing information in to query Stephen, I'll just ask a few questions. That one of the issues, as you pointed out near the beginning of your talk, is some of in some cases the plant did not, they were not able to, for a variety of reasons, score whether or not there was any colonization. There were all sorts of reasons why a plant might not be scored. So how did you handle the missing data when you did your Friedman analysis, your non-parametric Friedman analysis?
So what I did is, when I went in and so the missing time points I just didn't include because there wasn't any scoring data used, and any for some of my own data that had roots missing. Out of all of those time points, the majority missing was all in root time point 1, and there was only 4 that ended up being missed.
Okay.
So I was able to, with a sample size of about 60 samples, I was able to just, since there was no data there, I simply eliminated it. And when I performed the Friedmans, I went by the lowest number of repeats, which in this case ended up being 3, which allowed for a good analysis for all the different accessions.
Okay. And what you showed at the beginning that there were substantial differences between the different accessions. What are on colonization? What are some of your hypotheses about for explaining those results? I
believe the difference is based, while I cannot with confidence say certain things, I would hypothesize that the differences found were heavily based on I think that the colonization level, when it is growing with the plant, it reaches a sort of, apex and levels out. And so the variation, because of the massive difference between time point 2 and the mature being about, if I remember correctly, 5 weeks, that is a big growth time period. And so I think during that led to a heavy difference. And then the time point 1 difference being just the scoring method being a little more, giving out values that are more 0.1, 0.2, 0.4, 0.6, rather than the more varied values.
Okay. And I think the last question I have time for, did you have health indicator data on these plants? Like dry weight or height or anything like that you could go back and look at as far as the trade-off between parasitism and mutualism? Was there health data in that study?
Yes, there was. I got sent a whole file of health data with it.
Okay.
But at the time, I was trying to focus on the analyses, and there were certain elements where PIs were in the health data that weren't in the time point study, and vice versa. So there was a lot of not overlap.
Okay. So I'm going to have to stop there. Thank you very much for your work and sharing it with us. And now I'm going to move on to Matthew Jacoby. Matthew Jacoby comes to us from George Mason University, and he worked with Art Edison, but he also worked with Chris Esselman and Alex Molina, and so he had a lot of help, a lot of mentorship on this program, and so he's also going to be talking about Neurospora as well, and he'll be looking at metabolite libraries for Neurospora. Thank you, Matthew. Please go ahead.
Yeah, thank you so much, Dr. Arnold. As Dr. Arnold mentioned, I worked with Dr. Edison in trying to build a metabolite library for Neurospora, and I was using NMR and HPLC-MS. So a little bit of background about what I've been working on. Neurospora crassa, it's a well-studied modeled organism. It's known for the biological clock and its ease of lab growth in the lab, and its manipulation and introduction of genes and knockouts. So metabolomics and genomics, why are we studying metabolomics? Metabolomics, it's really closely related to genomics, as we see in the slide on the slide, the image. It shows genome, transcriptome, proteome, and metabolome. They're all within the same dogma. So genomics is really particularly studied among biologists, and metabolomics is just like a new and up-and-coming area of field. And so what Art's lab, or Dr. Edison's lab, works on is we work on NMR and mass spectrometry. So NMR, it gives us some information about the structure of our metabolites, whereas mass spectrometry, or MS, gives us information about the mass of the compound. The units are MBZ. A little bit about my project. So in a given cell, there are hundreds, maybe even thousands of metabolic pathways, and knowing which ones that particular cell or organism are using provides really substantial information about the organism, the environment it's in, and how we can go about treating any types of infections or anything. And so what we aim to do is we aim to grow our cells and extract our metabolites. These metabolites we can then perform 2D NMR on them, and we can get a baseline understanding of what types of metabolites are in the sample. And it can help us with identification further down the line when we do HPLC and mass spectrometry. So once we get to HPLC, we can fractionate and collect and concentrate each of those different metabolites into hopefully super small concentrated samples, which we then run mass spectrometry and NMR on them to basically understand what the compounds are and further develop like a repository or database which can help us with matching. And so this is all in aims to build up a baseline understanding for when we start introducing TOR mutants, or target of rapamycin mutants. And so when we get to that point, we'd want to be looking at differences between what the wild type and the TOR mutants looks like in the NMR and mass spec data. And so my data specifically highlights a particular strain, the QAX strain of Neurospora, which is a strain that was handed down by one of our collaborators. And so some of the major goals for my project, I wanted to work on optimizing their growth procedures, something that Alex and Chris noticed before I had entered into this program was that the older spore suspensions were growing more consistently, and they provided more consistent NMR results. And so I wanted to test a high concentration representing a new spore suspension and a low concentration representing an older spore suspension to see if there was any differences between the two. And there was another thing that they noticed, which was that when they were placing their tubes into the growth chamber, they noticed that the placement within the growth chamber often resulted in variability within the NMR results. So I also wanted to test how the placement within the growth chamber affected the results with the NMR. And I also, so after I'd done that, I wanted to look into establishing a basic understanding of like what metabolites were used by Neurospora. So we could do that through 2D sampling or 2D NMR results, and we then would put that into a database and look at that. And another part of my goals, understanding how the instrumentation works. As we know, nuclear magnetic resonance is something that's really difficult to understand. So trying to work with that and understand it has been one of my main goals for this summer. And again, after working with the NMR, I wanted to work with the HPLC and try and help them to optimize the procedure for fractionating. So as I stated a little bit before, my methodology, we want to grow and isolate the fungus. We want to freeze dry and extract our metabolites, concentrate our metabolites, and then we want to perform NMR and HPLC-MS. And this will help us to begin our fractionation process. I do have a link to a protocols.io at the end of this presentation, which takes you to a more in-depth protocol. Okay, a little bit more onto results. So when I say high inoculation concentration, I mean 10 million cells per milliliter versus low inoculation concentration or 1 million cells per milliliter. And again, when I talk about the placement within the growth chamber, I have left, front, back, and right, all labeled on the image. And so a little bit about our results. So right here we have an NMR spectra, 1D NMR spectra of my first batch of samples. First off, I want to note that their area between 4.5 and 5.5 ppm on the x-axis here, that is the region where water will show up. A lot of variability will occur here, and we should probably try to not analyze too deeply into this, and we want to make sure that we're interpreting this correctly. As we can see in our regions here, I've noticed that there are some asterisks. Those asterisks correspond to some interesting data where our results aren't particularly what we would want them to be. And so if I forward onto the next slide here, we can see a little bit more clearly all of these spectra. It appears that they have peaks within the same region. If we look between 2.6 and 2.8, we see some really consistent data with one large peak and two smaller peaks, coupled next to each other. And that's really consistent between each of the different samples that we had. I do want to point out that intensity of the peak is probably not what we want to be focusing on. That's dealing more with the concentration of our sample as opposed to just the peak itself. And so, again, looking at the asterisks, we see that they're, so looking at the asterisks, we can see that there's some really clear inconsistencies with how the spectra appear in comparison to the other main spectra. And so looking at that, we can see a little bit more closely our 0. So 0 here, we use a standard known as DSS, and this helps us for quality control for looking at the rest of our spectra. And as we note here, samples 008, 010, 019, and 025, all of the ones that I noted with the asterisks, they have some indication that there may be some issues with our DSS peak. And so this could be any variety of reasons, from incorrect shimming to not enough DSS to too much sample to even some particulate within our solution. And so first I want to point out that principal component analysis. So I wanted to look into how our data was varying. And I wanted to see how we could display that variation in a more easily accessible and easy to understand point of view. And so I did what's known as PCA, principal component analysis, which takes thousands to even millions of data points, and then it combines it into only a small subset of variability, and it allows us to analyze our data more easily. And so, as we can see here, I didn't remove out our outliers for this first one here, and we see that there's a really large variation within our first component. So we see hundreds of hundreds of points of variation within our first component. And so when we removed our outliers, we noticed that variation shrunk down a lot and significantly. So I think that was something really important to point out here. This next slide, I do have our placement within our growth chamber for the low concentration. And I do believe I have it for the high concentration as well, but I don't know if it will switch over correctly. But we can see that there are some significant variances, again, within our first component, which help us to explain that our outliers, so our outliers are significantly impacting how our data varies. And so moving forward, our decision, we decided to move the rack that was closest to the vent fan towards the center, which helped us to better control our data and make sure that we were experiencing less variability. We just wanted to make sure that our data was as consistent as possible. And so all of our future experiments were run using that center blue rack. Now onto our 2D experiments. So for 2D experiments, they tell us information about the interactions either between proton and proton or a proton and carbon-13. And that's particularly helpful for metabolomics because it can tell us a lot of information about the structure. And from these spectra, we can then input them into a database which could be matched to potential compounds. And so I only wanted to perform this 2D experiment on our pooled sample. And so this is our heteronuclear single quantum correlation, or HSQC, which was performed on our pooled sample, and our total correlation spectroscopy, or TOXI experiment. And so moving on to our database, we used COLMAR, which stands for Complex Mixture Analysis by NMR. And COLMAR helps us to match our spectra into a whole bunch of databases. It uses 4 different databases, which I've mentioned below, where we can upload our 2D spectra, and we can look at how the database attempts to match peaks within our experimental data to known peaks within the databases. And so right here, we have our data, as you can see in the red circles. Those are database peaks. Our green peaks are the matched experimental peaks. And then if we move over to the TOXI, you can see our expected cross peaks. And so once we go through that, we have to hand match all of our data to either be fair, good, or poor explanation as a matching for our compounds. And over here, I have assessed a short list of compounds that I believed were fair or good. Something I want to point out is pyruvic acid, which is known to be an intermediate in the citric acid cycle, which means that we're providing aerobic conditions, which was part of the specifications of our protocol. And finally, our HPLC MS, we want to look at the chromatography. Again, we weren't able to get to optimize and completely optimized spectra, but we want to further optimize our spectra moving forward. And so with HPLC, we can fractionate again based off of several characteristics like the mass coming off of the spectra, or even the UV vis spectra itself. And so our results, as I said, our results are preliminary stages, and we do not have any significant data to present at the moment. All right, so our next steps, again, continue to optimize our chromatography procedure so that our peaks are much more separated. We'll then move on towards collecting time-based fractions from the HPLC and do 1D NMR on those fractions. And after doing that, we'll collect we'll collect MS data or high resolution mass spectrometry data, and we'll get onto the fragmentation patterns. And once we get that, we'll begin to develop TOR mutant strains, which we can then use for comparison between wild type and TOR mutants. And finally, my acknowledgements. I'm sorry, I'm running a little over time, but I'd like to thank Dr. Edison for opening his facility. Again, Chris and Alex for helping me out this summer. Dr. Grimes and Ricardo Borges for helping me out with optimizing my procedures. And then again, Dr. Arnold for accepting me into this cohort and encouraging my research.
Wonderful talk. Looks like you're well on your way to getting the procedures for the fraction library. So again, people, you can, we'll have a few questions. You just have to enter at the bottom of your screen and I'll read them off. But while you're doing that, I'll just ask a question or two while you're doing that. So tell us why you picked TOR as an example of a gene in Neurospora that would be good to look at.
Yeah, so TOR, I believe, is known to regulate a lot of different kinases within the cells. And so if we look at TOR, we can try and examine a little bit more about what specific kinases are being infected and how the cell cycle is being maintained and regulated, which is really important when studying the clock, because we're assuming that there's a lot of kinases which are interacting with the different steps within the biological clock, and therefore by interacting and looking at different TOR mutants, we can specifically target different steps within the cell cycle.
Okay. So do we have some questions there? Or okay. So I'll just ask and make one more comment that it also has an important role in aging. So that's another, at least for some of us, that matters. So, so I guess I will go ahead and I just to stay on schedule, I want to thank you very much for a wonderful presentation, and it's exciting work, and thank you. So it's my pleasure to introduce our next speaker. Our next speaker is Katie Knight. She's from the Rochester Institute of Technology. Her mentor was Dr. Xiaoying Zhao. She focused on a bioinformatics problem and designing primers for potential immunotherapies, and I'm going to let her take over. Thank you, Katie. Please go ahead.
All right. Thank you, Dr. Arnold, for the introduction. So I was designing primers for T-cell receptor alpha chains this summer. So I've got some background about what my project is about. So first, I'm going to explain what T-cell receptors are. So T cells are a specific type of white blood cell, and they're part of the immune system. T-cell receptors are molecules that are on the surface of the cell that interact with and respond to antigen molecules. And they're also very specific to the antigen. They're made of alpha and beta chains, where each chain has a few extracellular regions. There's a variable, adjoining, and a constant region. There are some other chains as well, but these are the more prevalent ones. So we're working on TCR sequencing. The TCR alpha chain is generated by VJ combination, which creates the part of the receptor that contains all the variability, which then makes it specific to the antigen that it binds to. And this chain of proteins is coded for on the TRA gene, which is the T-cell receptor alpha locus. So we need to be able to replicate this gene in order to send it out for sequencing. So the way we do that is we use PCR, which is a method that lets you rapidly replicate a specified segment of DNA and requires the use of short synthetic DNA fragments called primers. But we're also working specifically with RHPCR, which is which is RNA's H-dependent PCR, which allows for the use of blocked primers, which can also allows for more specificity and less chance of primer dimers. So the intro to my project is I've been shadowing a PhD student. Her name is Jasmine D. Carter. She's already designed primers for the beta chain of the TCRs that we're working with, and I've been helping her optimize her protocol. So we've been working with changing the primer concentration, the annealing temperature, and also testing the RHPCR primers that she's designed. So these are all of my parameters for design. So we needed them all to we needed all of the primers to have roughly the same parameters, because eventually these are all going to go into a multiplex PCR, and it's much easier to run that if all of the primers are similar. So the primer size is 20 to 25 bases in length. We want the melting temperature between 60 and 62 degrees Celsius. The annealing temperature is 56 degrees Celsius because we want it to be 4 degrees below the melting temperature. Our GC content is between 40 and 60%. And the GC clamp/clap, which is not to exceed 3 C's and G's within the last 5 bases. We need to have at least 2, but if there are more than 2, then that can increase the melting temperature because there's a stronger bond between guanine and cytosine than between adenine and thymine. And about halfway through the program, there was a change in the parameters. So originally, we were designing the V gene primers 100 to 200 bases upstream of the VJ region, and the C gene primers about 50 bases downstream of the C region. But they were then changed to be much closer to the region that we wanted to capture. So instead of 100 to 200 bases away, we changed it to 40 to 70 bases away, and from 50 to 10 to 40. So the reason for the change is because we are interested in capturing the VJ region. And so when we move the primers closer, then we can ensure that we'll get the whole region and we'll have less extra stuff that we don't want. So the process that I used is I was given the DNA sequences from one of the other PhD students in the lab. So I would take the sequences that I was given from her, put them into primer3plus.com. I would input the parameters that I listed earlier, and the website would provide me with a list of several possible primers that I could use. Then I would then take those results and put them into Ensemble Blast, which would check for specificity. And we wanted to have it to have less hits on the genome that we were comparing it to, which would be the canine genome, because that means that there's a higher chance that it won't bond to anything that we don't want it to. I would then repeat these steps as needed until I got primers with a low hit number and the parameters that I wanted, and then I would write all of these down. I would write down the data or the information for the final selected primer that I went with, and I would then repeat this step for all of the genes that I needed to design primers for. So in addition to the regular primers, we also did RHPCR primers. They were kind of just additions to the primers that I already designed. It was a sequence of 6 or more. It is generally a sequence of 6 or more bases between the end of the primer and the beginning of the gene we want to capture. It doesn't perfectly match the base strand or the complement that it will bind to, because it has an RNA base that will correspond to the DNA base that it should have, and there's also a mismatch base at the end, and after that there's a C3 spacer, which is meant to block extension and prevent replication in later cycles that isn't supposed to happen. So here's an example of what that looks like. So I've got like the original primer, the like roughly 20 or so bases, and then the RH edition that's taken from then just like the next 6 bases in the sequence. And then after I make the adjustments, you denote the change from a DNA base to an RNA base with a lowercase r, and then the capital letter of the base that you want. And then the end, you change, instead of the DNA base that's supposed to be there, you'll denote the mismatch base with a lowercase letter instead. And then you put the whole thing together with the original primer, the RH edition, and the spacer. So I could not order and test my primers during the program time due to the change in parameters. So I don't quite have any results for that. As for why this is important, the well, first I'm going to talk about the future plans, which I don't think I have a slide for, but the primers will eventually be ordered probably soon, and then the protocol for them will be optimized in a similar fashion to the TRB primers that I was helping Jasmine test. And we can also test the RH editions if they're needed, but they may not be. And this is important because it can help us gain a better understanding of canine TCRs, because we're using the canines as a model for humans. This will also help us learn like what antigen cells have been exposed to, and it can also hopefully we can also hopefully use this knowledge to develop a cancer vaccine, because then we can possibly create cells that have T cell receptors for specific antigens that we want them to have. Then here are my references. And that's all I've got. Yeah, I'd also like to thank Jasmine, especially the PhD student that I was shadowing for most of the summer. And I'd also like to thank Dr. Zhao, my mentor, and Dr. Arnold for letting me be in this program. So,
Thank you for the wonderful talk. And again, audience, you can submit questions at the bottom of your screen, and I'll be glad to read them to Katie. So has there been any lab yet that's actually come up with, you might call a cancer vaccine, or are there any being tested either in the dog or humans at this time, or?
Not to my knowledge.
Okay, so it is a sort of totally new approach. So that's really interesting. And so you're presumably what you're going to do is not only looked at the alpha chain.
Yes.
But you're gonna look at other pieces of the receptor as well, and then hopefully down the road you'll have a sort of assay at a number of places in this gene that seem to be important. Is that the basic idea? Because you talked about multiplexing it.
There are, since the one end is a variable region, that just means that there's a bunch of different versions of that part of the sequence. So the multiplex PCR would apply to just taking like the DNA sequences and then putting all of the primers in to get the different versions all at once.
Oh, okay. So you on one patient, you'd be able to see all the different receptors but rearrangements that have been generated by the immune system, and hopefully gives you some idea of targets as well. That's okay. That sounds great. So I think I'm going to, let's see if there's any questions. I don't see any questions and so what I will do is we're right on schedule, and thank you very much. And we will move to our next speaker. Our next speaker is Mariana Labat. And I'm going to give her a brief introduction. Mariana comes to us from Case Western. She has actually had two mentors during her project. One is Shrina Pradhan, and she's worked very closely with Shrina both on the molecular biology as well as the bioinformatics, so that Mariana could achieve her goal of getting true systems biology training. And she has also benefited by the mentorship of Tom Pendergast, and both of those individuals are in Katrine Devos's lab. And she's picked a really interesting problem from the standpoint of UGA's entrance into genetics through the work of Dr. Burton, who gave us the zoysia varieties that we have on all golf courses around the United States. And so she, and that is very important for Georgia in particular. So she's going to talk to us about pigments, anthocyanins, in at least 3 different zoysia species. And please take over, Mariana.
Yes, thank you so much for the introduction. So this is my presentation, and it's titled Molecular and Computational Analysis of Anthocyanin Regulating Genes in Zoysia Grass. Now, as you can probably tell from the title, there are 2 major portions or categories to this project. The first is a molecular approach, and the other is a computational, more comparative genomic approach. So I'd like to start my presentation by going over the molecular approach we took to this problem. So to give you a little introduction, so zoysia is a type of salt-tolerant turf grass, and there are 3 main species. There's Japonica, Matrella, and Pacifica. So these are the 3 main species that we're going to look at throughout the project. And although it's a salt-tolerant turf grass, the trait that we're particularly interested in is anthocyanin content. So anthocyanin is a chemical compound strongly associated with red and purple pigment in various plants. And in the zoysia grasses specifically, it produces purple flowers, which you can see in the image on this slide. And why are we interested in understanding anthocyanin content? Well, we want to better understand the biosynthesis of anthocyanin for various reasons. First is food manufacturing. Many food manufacturers want to replicate this pigment in foods, and in order to do that, you have to understand how it's synthesized and the genetics behind it really well. And also plant breeders who breed grass and want to create more uniform looking grass are also going to want to be aware of this particular trait. Also, zoysia is an orphan crop, so not many people study it. So we thought it was significant that we try to expand our scientific knowledge of this particular grass. So before I go further, I want to talk about the difference between an F2 and an F1 population, mostly because these are terms I'm going to be throwing around a lot. So an F1 population is a cross between Zoysia japonica and Zoysia matrella in our case, and then an F2 population is a member that we've taken from F1, and we have selfed it, which is kind of like a form of self-pollination. And if you look at the graph on my slide, you'll see that at least in the F2 population, there's a higher prevalence of anthocyanin presence than absent in this particular population. So in terms of what the genetics behind this is, what was noticed is that we were looking at a candidate gene was looked at. And in the case of purple plants, what they noticed was that when there's a G at a particular position, the intron is properly spliced out and the gene is able to be expressed correctly, and you get a purple plant. However, when that G is turned to an A, the introns aren't properly spliced out and we get no gene expression, and this leads to a green plant. So this led us to our question because the phenomenon I pointed out in the previous slide was what was noticed in an F2 population. So that led us to the question of, is the same locus responsible for anthocyanin in F2 and in F1? So basically, are the genetics behind anthocyanin in the F2 population working the same in the F1 population? So the way we went about doing this is we collected leaf tissue from 32 Zoysia japonica and Matrella plants, or 32 F1 plants, which are a cross between japonica and Matrella. We extracted DNA from each tissue sample, and then we amplified our candidate gene or our gene of interest using PCR. And then we used a restriction enzyme digest, which basically cuts our gene of interest if the SNP is not there. So you would see a cut if the plant is purple or phenotypically purple. So here's like our expectation versus our results. So we expected to see something similar across F2 and F1 plants. So if you look at the gel labeled F2 plants, there's a solid band for a green plant, which is indicative of the restriction enzyme not cutting because the SNP is there, whereas you see a cut for purple plants or multiple bands because it's either homozygous or heterozygous. Now, if you compare our F2 plants to our F1 plants, you'll see some drastic differences. So if you look at the green arrow and the purple arrow in F1 plants, the bands look identical, or the band pattern looks identical, and the one at the bottom looks really different as well. So this is pretty indicative of like there being big difference between F1 population and F2 population. So from this, we were able to conclude that the locus in F2 plants that's contributing to the purple flower phenotype is not the same locus in the F1 plants that's contributing to their purple flower phenotype. And we're in the process of further studying that. I wish I had results to show you, but science is a slow process. And now I'm going to move on to our comparative genomics approach, which is also our more computational approach. So to give you a little background to that approach as well, you might be able to guess from the term comparative genomics, but we compared Zoysia to another plant species, and that species is finger millet. So the Zoysia grasses are a member of a subfamily called Chloridoideae, and finger millet is also a member of the subfamily, which is part of the reason why we chose it for this side of the study. And also, it's better studied than the Zoysia grasses, so there are more resources that we can use available online and available to us. So the broad goal of this side of the project was to use finger millet genetics to get a broader understanding of anthocyanin and evolution in Zoysia. So how we went about this is there was a lot of trial and error, but I think that's why I loved this part of the project the most. So we took 33 known anthocyanin-related genes in finger millet, and we blasted that against the Zoysia genomes. From that blast result, we were able to get subject alignment data and scaffold data, and we were able to feed this information into a Python script to generate a gene prediction. And we did this for a random 5 out of the 33, and we did this for each genome, so Japonica, Matrella, and Pacifica. So from there, we went on to protein prediction, and we originally tried to use the Zoysia genome database, but we quickly realized that there are errors in the genome database and in their predicted peptide sequences. So we switched to FGene SH+, which is a hidden Markov model. And it basically uses a nucleotide sequence, which in our case is our gene for our gene prediction. And then it uses a peptide sequence from a related species. So we used a finger millet peptide sequence. And with those two combined, it's able to predict what the protein should look like, or what the protein sequence should look like in Japonica, Matrella, and Pacifica. We also ran into issues with here with the finger millet peptide sequences being accurate. And this is largely because the annotations are often automated. So so issues come about. And we wanted to confirm that the peptide sequences are real. So we used an alignment to guarantee that they're functional. And then we also used IGV with RNA reads to confirm that these proteins do exist and are actually functional. I would go through the process of like how I checked on IGV, but I'm kind of running low on time, so I'm gonna skip to the phylogenetic trees that I generated for this project. So I used my alignments for my protein predictions to generate my phylogenetic trees. And this is one of the set of finger millet genes that I use to create my protein predictions. And what you're going to notice quickly in here is that there are two main clusters, and that's pretty indicative of two subgenomes, because Zoysia is tetraploid, meaning it has 4 sets of chromosomes, and two subgenomes, usually referred to as an A subgenome and a B subgenome. And if you look, finger millet is in a cluster with that is almost looks like one of our subgenomes. So it almost looks as if finger millet is more closely related to one subgenome than the other. And we saw something similar in other genes as well. And this gene, again, you see it where finger millet is more closely related to one subgenome than the other. So we wanted to guarantee that this pattern is something that we are actually seeing. So we added in the species Ceteria italica and Orpedium thomeum. And what you'll notice is that once we added these two other species, finger millet is removed from one of those clusters. And this is more representative of what we would actually see or what we would expect. But what you're going to quickly notice is that what stays consistent across the trees is this particular patterning where there's a japonica gene related with a Matrella gene, or a protein, or a Matrella protein with Pacifica protein, and that stays consistent across. And it caused us to realize that the Zoysia Matrella is actually a hybrid between Pacifica and japonica, which is why even over here, when we try to replicate this for single copy genes in Ceteria, you see a japonica protein with Matrella protein. So it kind of further confirmed what we were thinking. And then here are the blast hit patterns for the anthocyanin and the single copy genes that I did. And I wanted to point this out because Zoysia is a tetraploid. So there's an A subgenome and a B subgenome. So in theory, you would expect there to be 2 hits, one for each subgenome. But really, we don't see that. We see 2 hits, 4 hits, 1 hit, 3 hits sometimes. And that's also similar in the single copy genes. And part of the reason we think this occurs is because Zoysia is a much older tetraploid than something like finger millet. And the older the tetraploid, the more likely they are to lose copies of genes because it's not particularly advantageous to have them. And that is all for my presentation. I'd like to give a big thanks to the DeVos Lab, especially my co-mentors, Shrina and Tom. I couldn't have done it without y'all, and thank you for such an amazing summer.
Well, thank you very much, Mariana. It was a wonderful, wonderful talk. And so do you, again, people, you can go to the bottom and ask, type in your questions and I'll read them to Mariana. And so I wanted to ask, sort of on the first part of the talk, where you've got this very interesting difference between the F2s and the F1s. What is the next step to look at that?
Yeah, so I was working on the next step even yesterday. So we have another candidate gene that we're interested in, that has also another SNP that we're interested in that we think might be contributing to the phenotype in the F1s. So I did my PCR for the 32 F1s yesterday, or I finished it up, and now it's Shrina's job to sequence it and see what we get. So I'm hopeful.
Okay. And so the fact that these anthocyanins seem to be like they seem to be in a multi-gene family, it's kind of complicated because you've got a tetraploid and on top of that as well. So you said there were like 33 members of that family if you looked across the different species. Am I getting that right?
So actually there are 33 different genes that we looked at, but they're across various different families because anthocyanin biosynthesis is just so complicated that it uses a lot of different genes for the process to work. So we have MYB genes, we have flavonoid genes, so it's really a variety.
Right. Okay. We hit our time, so I'm going to have to move. Oh, we have one question. So okay, I'm going to invade our next speaker's time with one question, and it's going to come up in a second. Oh, here it is. Excellent talk, Mariana. Quick question: could the majority green coloration in the F1s be due to heterozygosity that becomes fixed in the F2s? That's an anonymous question.
Is that majority green coloration? So, I'm not sure I quite understand this question, but I don't think there's necessarily a majority of green coloration in the F1s.
Yeah.
I think they might be getting confused about how the bands look, though all those bands are not green. Some of them are purple and green. The ones I pointed out are just different. And it looks like in our case at least that like at least the top row of the gel is very much heterozygous for the SNP, where some alleles have it, some don't. And that's why we see the top band and the two bottom bands. But there really is no like super consistency when you look at the F1s.
Okay. Well, thank you very much. And I'm going to move on now to our next speaker. And it's my pleasure to introduce Arlene Martinez. She comes to us from the Florida Gulf Coast University, just south of us. And her mentor was Mrs. Shufan Zhang now. And she's going to talk. Her project is part of the AMF Sorghum project, headed up by Jeff Bennetson. And she's going to talk about how we actually detect these arbuscular mycorrhizal fungi once we have these beautiful images. And she's going to talk about a neural network to do that. So it's my pleasure to have you here today. And please go ahead, Arlene.
Thank you for that introduction, Dr. Arnold. I just want to preface, I am a little bit under the weather. So if I cough or sneeze, just excuse me. So my project is on object detection using mask-RCNN. Before I get into what exactly mask-RCNN is, I'm going to go into the background of why we're doing what we're doing. So AMF is a soil fungi, and they establish a symbiotic relationship with most plants. So when that symbiotic relationship is established, spores present in the soil will germinate and infect the root system of the plant and form arbuscules in the plant cell. The arbuscules are sites for nutrient exchange, which is one of like the aspects of the symbiotic relationship between the fungi and the plant. And the way that we determine whether there's an established relationship between the plant and the fungi is by looking at the images of the root structures. So we'll look at things like the hyphae and the vesicles and the arbuscules, which is what I focus mostly on in my project for the AI object detection. But if we were to, since we like sample a lot of root images, it would take a lot of time to go through each image individually and like identify how many, like what's going on with like the arbuscules and the vesicles, because the images are so dense and full of so many structures. So the simple solution to that is AI or mask-R-CNN. So a preface of mask-R-CNN. The CNN part is a convolutional neural network. It's a simple artificial neural network used for image recognition and processing. So a convolutional neural network is much simpler compared to like a region-based convolutional neural network, only because so the convolutional neural network can only detect one object in one image. So it can't do so much, it can't do as much as like mask-R-CNN does in terms of like looking at images that have a lot more going on, like the images that we use for like the mask-R-CNN and like training the mask-R-CNN. So another thing that I wanted to mention is that mask-R-CNN is based off of another type of AI model known as faster RCNN, which basically it basically takes different features from an image all at once, and it creates like it like outputs everything into a single output. And it's called fast RCNN because it literally has a fast computation speed. So it's able to like retrieve and interpret instructions faster compared to like another type of AI model. But mask-R-CNN itself is a type of segmentation on objects that works at a pixelated level, and it detects objects at like a pixel level. So the way that mask-R-CNN is broken down is into two types of image segmentation. Image segmentation is basically, it breaks down images into multiple segments, and each segment is like analyzed. And then like back into what mask-R-CNN is and like the two types of image segmentation. So we have semantic segmentation and instance segmentation. So semantic segmentation doesn't have as much precise object separation like instance segmentation does. And as you can see, for semantic segmentation, the only thing that's separated is really the object from the background, whereas instance segmentation has a more precise outline for each object, and it's each individual object, or in this example, each person is separated from the background. And it's just overall better. You can also see that it adds like a mask over it, which also helps with the precision and the object detection of the image. So the hypothesis that we went into this experiment was that okay, let me preface that this AI model, mask-R-CNN, has been used in the past for other parts of like the structure. So things like it's been used for like the hyphae and like the vesicles, but the object detection for arbuscules isn't as good as it can be. It's not as accurate. So we were hoping to improve that in this experiment. So the way that we were going to do that is through creating two separate labels, two separate groups. So singular arbuscules and clustered arbuscules. And hopefully by separating the two like types of arbuscules, it will increase the object detection. This is kind of an example of what I'm talking about. So this is what I did kind of a majority of my project. I individually annotated each arbuscule, and as you can see, there's like singular arbuscules and then there's a clustered arbuscule. This is kind of more, this is like a simple, this isn't super dense, but this is the overall look of what one annotation looks like. And it's this, like I did this 10 times. So it was a very time consuming process, but it was fun. And then just so that you could see, for 184, this is like what I would consider a cluster arbuscule. And for like image or annotation 2, that's like what I would consider a singular arbuscule. So the experimental design, we used a software called BGG Annotator, and basically I just went in and clicked around a bunch of arbuscules and then labeled it. Each image was annotated by hand. Again, it was a very time-consuming process, but the way that I followed the annotations, the way that I kept it consistent was by labeling, like having regulations for the way that I was labeling them. So for clustered arbuscules, if there were 2 or more arbuscules with no clear outline and they just were kind of all bunched up together, I labeled that as a clustered arbuscule. And then arbuscules that were singular and had a clear outline, as clear as an arbuscule can be, I would label that a singular arbuscule. And then I would exclude arbuscules that were too faint, because the AI model is literally taking in what you're literally feeding the AI model. So if you're feeding it incorrect information, you don't want to mess it up in terms of like, it literally, like, think of it as like it literally takes in the image and it'll try to replicate it, and I didn't want it to like confuse it for other structures that might look similar to like a faint arbuscule. And so then once the AI model was, once the images were annotated, I had to train the AI model and then test it and then inference it. But due to time constraints, I was only able to annotate all the images. However, I am continuing this project when I go back home. So I'm currently working on that process of training the AI model, which includes a lot of coding. And I have very little coding experience, but I do like the process of like going through and reading code and like learning and getting better at it. So I didn't want to rush the last process just to get results. I wanted to enjoy the process and take my time with it, and also just get good results. I want to see good results at the end of this project. So fingers crossed it goes well. But the way that we're going about training the AI model is, so basically we could have resized the image first and then tiled it. So basically what tiling an image is adding a grid over the image and then like cutting it into multiple pieces so that the AI model could take in each piece. And like the reason why we did that is because there are more singular arbuscules than clustered arbuscules. And that is an issue because you want to have an equal amount of labels and an equal amount of like annotations to feed the AI model. And like if we only feed the AI model singular arbuscules, it's only going to be able to detect singular arbuscules very well. So we decided to segment the images so that there could be a more like, we could pick and choose which like clustered arbuscule and singular arbuscule we wanted to feed into the AI model. And then the testing part of the AI model is literally taking one of the images from like the 15 total that I had and like feed it to the AI model, see how well it is at detecting the arbuscules. And then wait, seeing how well it is at detecting the object detection for the arbuscules, and then hopefully The inferencing part is where we go in and try to predict what happened, what we can improve in future experiments. And then hopefully the expected outcome is that labeling the singular arbuscules and the cluster arbuscules as two separate categories will improve the object detection. That's what we're hoping. We'll see. But it's going to be a long process, and I'm excited to continue my project when I get back home too. So it'll be something fun to look forward to. And these are my references.
Thank you so much. That's really, and I'm sorry you're under the weather.
It's okay.
And so this is an exciting project, and hopefully it will actually lead to a big improvement in the performance of the RCNN. So, the scripts that you're using, and I should say that please again go to the bottom of the screen if you've got questions for Arlene. Just type them in and I'll read them. And so the Python scripts that you're playing with, first of all, they are Python scripts.
Yes.
Yes.
The tiling image one is Python, but I think one of them was in R, and I was a little bit confused because I don't work with R.
Okay. And so we do have a question, and the other person's gonna pop it up here for me to read. There it goes. Okay. This is a question, it's anonymous. Can you explain why it is necessary to resize each image rather than feeding the entire image directly to mask RCNN? Thank you. That's the question.
Oh, that's a good question. Okay. So actually, there is an optimal size image for mask RCNN. And the problem is that if the image is too large or too small, the object detection is going to be off. So you want to resize the image for that reason. That's a good question. Thank you.
Right. So now the images that you used, and I'll take any more questions. I'm just filling the gap here. But I guess I'm invading the next speaker's time. But do you think, if you took the robot that generates the images, would your job of resizing the images be made a little easier, do you think, based on the way that you prepared the images for microscopy? Is that?
I mean, I wouldn't say that it's I don't know exactly how it would affect the imaging, because it's already included in the code. So it's kind of like,
right.
I don't feel like it would affect the way that we resize them anyway.
Okay. So what I will do is I'll just thank you for a wonderful talk, and we look forward to continuing to work with you and hope you feel better. And I'm going to go to our next speaker right now. So it's my pleasure to introduce Cole Nesselson. He's, like Arlene, is working on the AMF sorghum project that's led by Jeff Bennetson, and he's comes to us from Brown University, and he is actually following up on an idea that Phil Braley and Jeff's lab has been suggesting we pursue for some long time, and that is to have an independent molecular validation of the RCNN method for segmentation that Arlene was just talking about. So I'm just going to turn it, and he's also at the same time going to relate his findings back to Easton and Sedona's and Nancy Johnson's work looking at the heritability of AMF colonization. So please take over, and look forward to hearing you.
Thank you so much, Dr. Arnold. So yeah, my summer research focused on analyzing the root microbiome of sorghum to assess both the arbuscular mycorrhizal fungi community, the different species, as well as their abundance in sorghum's roots. So first, just a bit of background on arbuscular mycorrhizal fungi. They're fungi that symbiotically associate with 80% of land plants, and they provide nutrients such as phosphorus primarily, and sometimes nitrogen in exchange for the plant's carbon. And not only do they provide enhanced access to these nutrients, they also enable plants to be more drought tolerant and disease resistant. And also here I've indicated with the red arrows several arbuscules, which are actually the site of nutrient exchange. They're a very high surface area region where nutrients transfer between plant and fungi. And here with the blue arrows are the extravadical mycelium that are the majority of the fungal biomass in the soil that engage in uptake of nutrients and then transport to the host plant. And so we research AMF and their symbiosis with plants because they offer an ability to eliminate the need for energy-intensive inputs like phosphorus and nitrogen fertilizers. And in the case of sorghum, as a biofuel crop, it makes it a more carbon neutral form of fuel because you don't need to use much energy in providing fertilizer application and that sort of thing for the sorghum. Also, with climate change, AMF symbioses provide plants more tolerance to abiotic stresses like higher temperatures and biotic ones as well, with pest problem exacerbations as well as disease problems. Now, so for my research specifically, I dealt with the analysis of metagenomic DNA samples gathered from a population of sorghum. This population was made up of 2 parents and 15 offspring accessions of sorghum. And in my first experiment, I looked at the AMF community by sending out amplicons for sequencing. And so once I get those back, I'll have results on the differences in AMF species composition in each of the accession's roots. And it's important work because in the future, we'll have breeders that are focusing on breeding plants that are more symbiotically their association with AMF will be optimized based on this heritability measure. And my second experiment was more geared towards quantifying the abundance of AMF in each of the sorghum's roots using quantitative PCR and microscopy. So with quantitative PCR, you get a measure molecularly of how much the abundance of the AMF nucleic acid in each of the plants. And with microscopy, we can compare we can analyze the number of arbuscules in each of the slides, in each of the root samples, and we can compare those 2 methods of quantification. Like Dr. Arnold mentioned, though, it's cool because Arlene is working on this new cutting-edge mask RCNN algorithm. And so it'll be interesting to see how the molecular means of quantification with qPCR matches up against that new technique to quantify AMF in sorghum roots. Now, first, we conducted a field experiment on 15 accessions of the offspring accessions of 2 parents. These parents were crossed, and then their offspring, each of them were selfed for 5 generations, meaning that the resulting accessions are completely homozygous for all their traits. So in the greenhouse, the plants were grown for 4 weeks before their first planting. And then 2 weeks later, they were harvested. So these are relatively young sorghum plants, but they still have pretty interesting AMF characteristics, as you'll see. So after harvesting the AMF plants, the sorghum plants, we extracted the metagenomic samples of DNA by first grinding the roots with liquid nitrogen to homogenize them, and then using CTAB buffer, which is a surfactant that sort of disrupts the bilipid membrane of each cell, allowing the nucleic acids to escape. And then to purify that new mixture to purify the DNA from other contaminants like protein and polysaccharides, I used a phenylchloroform extraction. The phenol is able to denature the proteins and bring them down to the organic phase that is made more dense by the chloroform. And so now we have the aqueous phase containing the DNA and the organic phase containing the majority of the contaminants. Now, to precipitate the DNA out of the aqueous phase, I used ethanol because it dehydrates the DNA, which is a very polar molecule. And then once the DNA is dehydrated, it's able to precipitate out with the salt that was in the CTAB buffer solution. And in troubleshooting this protocol this summer, I was able to find that using ethanol is actually yields a more pure DNA than isopropanol, even though it's slightly worse at dehydrating the DNA. So it's interesting, and isopropanol it's much more efficient at precipitating out the DNA, but also can precipitate out unwanted chemicals and molecules like polysaccharides and that sort of thing. So after you precipitate, I washed with ethanol, 70% ethanol this time, to remove excess salt and other contaminants, and then dissolved in a high or low salt TE buffer, which is a tris EDTA buffer with a pH of 8. And that stabilizes the DNA. So after extracting the DNA, I amplified a specific part of the AMF genome in the D2 region of the large subunit part of the ribosomal RNA gene. Now, this particular gene, it's useful to use as a method to distinguish species of AMF, because within a species, it won't vary so much. It's conserved, this region in the DNA, but between different species, you'll have slight variation. So it's a method to barcode different species of AMF. There are other options to use from this ribosomal RNA gene. So like the ITS region, it's more conserved in general, and so it serves a better way to quantify the broader fungal community. And the opposite can be said about the small subunit, which is very, very much more variable. And even within a species, you'll see variation. So it's not great at distinguishing at the species level. And so let me be specific. This was my first experiment about trying to characterize the different species of AMF. Now for my second experiment was geared towards measuring the abundance of AMF in the root samples. And this, I used quantitative PCR, which uses a fluorescent dye that when incorporated into newly formed double-stranded DNA during PCR, during the elongation phase of PCR, it excites the fluorescent molecule, and the machine will be able to see this fluorescence progress as the polymerase amplifies more and more DNA. So the larger the starting concentration of DNA, the sooner the machine will recognize the fluorescence of the sample exceeding a certain threshold. So that's how we're able to quantify based on the cycle threshold. And with microscopy, I made slides and of small root fragments to be imaged by the high throughput microscope. And then by hand, you're able to see if a region has arbuscules and then deduce the percent colonization of AMF visually. So when it comes to my harvest data, you see the height data is very regular, even across different measures like stem length and base to longest leaf. And the very low p-value means that after running the ANOVA test, it means that it's very statistically significant that the height is variable based on the genotype. And the same can be said about biomass, a very statistically significant result. Now for my DNA, we got very relatively pure DNA at the correct 260/280 ratio of 1.7 to 1.95, and a good yield as well, as from 50 to 250 nanograms, and that's with 100 microliters of elution buffer. So plenty of material to work with. Here's a gel of my DNA, and you can see based on this ladder on the left and right side, the DNA is very high molecular weight, more than 1 kilobase fragments. So that's good for downstream applications like PCR and sequencing. And here we have the endpoint PCR amplicons that I generated for sequencing to characterize the AMF communities. And you see the bands here mean that there are very that almost all of the DNA samples that we got have presence of AMF. And so now for my qPCR experiment, first I had to calculate the primer efficiency of this AMF gene. Now this is actually a different. It generated a different amplicon, the small subunit amplicon. But its application different. The AM fungal. Sorry about that. The AMF gene was, or the AMF. Sorry, the primer was a slightly variable in terms of its efficiency, whereas the plant primer that I referenced to was much more regular. So I'll just skip ahead a little bit. So we have two ways of quantifying or of understanding our results. One is the delta-delta CT method, which has a much more statistically significant result. And it's interesting though, after running a post hoc test to see which samples cause and how they relate and how they cause variance, you see two samples at the bottom of the screen really contributing to the most variation. And so that calls into question a little bit of the heritability measure that I got was 0.81, which is quite close to 1, which suggests that MS is quite heritable, the abundance that is. But yeah, in terms of correlating microscopy with qPCR, I was only able to get a little bit of data, but you see the beginnings of a positive correlation here. So in the near future, I'm going to continue working after I get my sequence back and characterizing the AMF community, as well as comparing Arlene's MassCAR CNN with this qPCR data. So I just want to briefly say thank you to Dr. Arnold and Dr. Braley Jones, who helped me a lot with amplicon generation and statistical analysis, and Dr. Shida for helping me with the DNA work and troubleshooting that, and Mr. Lance for helping me with getting my microscopy images, and Isabella for making slides and helping me with the microscope as well, and Stephen and Anna and Arlene for all helping me make slides. Thank you so much.
Thank you for the wonderful presentation. And it's so nice to see all these projects coming together. And I'll take, if it's okay with Danai, I'll just take one question because we're invading her time. So if someone wants to put something in the chat, I'm giving you a chance, or right at the button at the bottom. And okay, so I'll hold off, and wonderful talk, and it's a wonderful data, and I'm sure people will hunt you down. Okay.
Sounds good.
Okay, so I'm going to turn to Danaya now, Danaya Robinson, and thank you for allowing Cole just to continue a little longer into your time. So Danaya comes from us to us from. You can't hear me?
No, go ahead.
Okay. It comes to us from Clark Atlanta University, Her mentor is Michael Turns, and she's going to be talking to us about the CRISPR-Cas system and a new type 4 system for CRISPR-Cas. Please take over, Danaya.
Yes, thank you, Dr. Arnold, for the introduction. This summer, I spent most of my time working on uncharacterized type 4C complexes. Hello, everyone. As stated, my name is Danaya Robinson. I am a junior biology major attending Clark Atlanta University. I would like to thank the REU program for giving me the opportunity to present what I have worked on this summer. Sorry about that. All right, I'm going to give a brief overview of CRISPR-Cas. So CRISPR-Cas systems are adaptive immune systems for bacteria and archaea. CRISPR-Cas systems recognize pieces of a virus or plasmid DNA when it enters the cell. The system works in 3 stages. The first stage is adaptation, which is a set of specific proteins that recognize short segments of DNA from the invader. They take it and integrate the DNA. It determines the CRISPR element in CRISPR-Cas. Second, in a CRISPR RNA biogenesis, the whole array are transcribed into RNA. Special enzymes come in and break the array up in smaller segments known as CRISPR RNA. Finally, interference, the proteins that were turned Cas form the scaffold around the RNA, and then once the same virus or plasmid re-enter the cell, the RNA in the complex complements the invader and binds to the section of the matching section of the invader. This can either be a DNA or RNA cleaver destroying the invader. Next, I'm going to go into the CRISPR system. As you can see, are broken down into 2 classes. Class 1 are multiple subunits, so they have 4 to 6 proteins that compose them. Class 2, they function as a single protein bind to the RNA. Connor and I mostly worked on these type 4 CRISPR-Cas systems. Our job was mainly focusing on purifying the proteins and then trying to determine if they are forming multiple subunit complexes around the RNA. The function of these systems are well studied, and the type 4 are still uncharacterized. Okay, so when looking at these models of the type 4C, this is what the complex might look like. We believe it resembles a type 3 complex. CRISPR RNA is necessary to form complex. Cas10 is the functional nucleus subunit. And the CRISPR RNA, the 5' tag, is what we're mostly working on. That defines the specificity for the RNA. Protein Cas7 and 11 are the backbone for the complex 5, recognizes the 5' tag, and then allows the complex to form. Next, I'm going to go into the organism information. So here I'll be talking about one of the organisms we use for characterization. I would like to emphasize a hypothermophilic aspect of Pyrococcus. It also relates to the protein purification. Pyrococcus is isolated from an hypothermal vent in the ocean and just one of the host encoded CRISPR arrays. So this is a question we wanted to answer this summer. Do type 4C complexes associate with a CRISPR RNA in a 5' tag specific manner? Now I'm going to go more in depth of the protein purification. Here you can see the protein purification I was able to have hands-on experience with. The plasmids in CRISPR RNA are transferred. E. coli arabinose turn on production, and specific amount of protein are put into the sonicator, and the sonicator uses ultrasonic waves to burst open the cells and release protein. The protein binds due to the interaction between histidine and the nickel metal. After the process of the heat precipitation, it is on to the centrifuge, excuse me, and separates the out native E. coli proteins. We fill the plastic columns with nickel and place his-tag proteins. Imidazole is used to compete with histidine and elute proteins of interest. Now you can see our protein purification data. So you can see that the Cas10, this is where we expected, as well as the Cas5 and Cas11. You notice the bands in these corresponding areas. You may also notice the slide D accompanied by the asterisk, and it is just a protein involved in the complex. It is a common nickel binding protein that contributes. We were able to co-purify these complexes by pulling on the single tag subunit. And E. coli, the CRISPR RNA is not required for them to form to this level, but is required for function. The arrows indicate these individual proteins of the type 4 complexes, and these identities were confirmed by mass spec. Here, I will give a brief explanation of extraction. So the RNA protein samples are treated with hence trizol. You can see here. It stands for phenol and chloroform and guanidinium. That separates into 3 layers containing DNA, RNA, and proteins. We take the RNA layer and use ethanol to pellet the RNA. Then we take the RNA and run it on a urea page gel. Now we're going to go into the RNA data. As you can see by the gels in the positive control, you can see a band is being expressed. It shows no CRISPR RNA band as 67 non-target and no CRISPR RNA in mutant samples. In the sample with wild type, we see the CRISPR RNA band. This confirms that the binding does appear 5' tag specific. The mutant CRISPR RNA is just a complementary sequence to the wild type. To further explain, this mutation was shown to inactivate CRISPR RNA binding. So here are conclusions. Pulling on a single tag subunit returns complete 4C complexes. 4C complexes coalute with a supply native CRISPR RNA. CRISPR RNA binding by the type 4C complexes is a 5' tag specific. And our next steps would be moving into native systems to confirm our results. Finally, I would like to acknowledge the REU program, Dr. Arnold, Dr. Turns, the Turns Lab. Most importantly, a big thank you to Connor Pittman, my in-lab mentor, for his utmost patience and for giving me the opportunity to work on his project. And thank you.
Wonderful talk. You really you addressed the question. You got to the end point. That's wonderful. So and again, anybody who wants to ask questions can enter in at the bottom of your screen. You should be able to send in questions, and I'll just act as a space filler question for Danaya. Do the type 4 systems provide any sort of functionality that the other systems don't? What's driving the interest in these type 4 systems?
So these type 4 systems are mostly uncharacterized regarding structure and function. These systems are intriguing for 2 reasons. They are frequently encoded on mobile genetic elements as opposed to in genomes. They rarely associate with a linked CRISPR array, leaving the source of their CRISPR RNA a mystery among the type 4 systems. And the type 4C are a very promising option for our lab.
Oh, okay. That's very, very neat. Okay. Here's a question for you. Great presentation, very informative. What are the advantages of CRISPR? That's from an anonymous person.
Good question. CRISPR-Cas are similar to a human immune system. They're adaptive. So we have antibodies in our immune system that will recognize pieces of a virus in a bacteria and it will respond. So the advantages you ask is important because it is used to edit and treat genetic diseases.
Oh, wonderful. Okay. So I'm going to we're actually back. Are there any more questions out there? Okay, we're going to stop there and we're back on schedule with our last speaker. And thank you so much, Danaya. Wonderful job. And we'll now move to our last speaker before the lunch break. And it's my pleasure to introduce Jake Spittle. He's from the University of Georgia. His mentor is Isaac Torres, and he will be talking about another feature of the AMF sorghum project. He's going to actually be looking at data from the Arizona experiments. And I'm just going to, ultimately, he's going to compare that data with similar experiments that were done last year in Georgia. So Jake, go ahead, take it away.
Thank you for the introduction, Dr. Arnold. So my project is titled Sorghum Bicolor GWAS on Height Comparing BAP Accessions in Arizona versus Georgia. And so for some background, sorghum is a heat-tolerant grass species that is cultivated for biofuel or its grain for food. It's like corn, but it's taller and more heat-tolerant. It is also a monoorganism like corn, and it can thrive in less fertile soils than it. Sorghum is known to have a mutually beneficial association with arbuscular mycorrhizal fungi, abbreviated AMF. And this AMF give plants phosphorus and sometimes nitrogen in exchange for carbon, which is all the carbon they receive is from the plant. And they live inside its roots. And AMF are also known to increase water absorption as well as disease resistance. So for the overarching project, it is an attempt to study the relationship between sorghum bicolor and AMF to find quantitative trait loci that have the most beneficial associations with AMF. And we believe that it can be used as a highly effective biofertilizer for sorghum to increase the biomass, given that we find the right associations. And we are attempting to quantify this through high throughput microscope imaging to determine the percent colonization of AMF of these different genetic accessions. And I'd also like to point out that as part of the project design, there are 4 different treatment blocks, one with low nitrogen phosphorus, one with low of one, and then high of the other, and then both highs and both lows. And then, so this is an example of one of the images that we got from our microscope. So in the dark gray, you have the root, and the light gray around it is the cortex. And the blue within that is the AMF that we have stained so that it is easier to see. So this is how we are quantifying the percent colonization. And then, so for my individual GWAS, I focused on the height differences between our sorghum here in Georgia last year and the sorghum just recently harvested this summer at northern Arizona. And so like the overarching project, I'm looking for QTLs that are significant, but in this case, I'm doing it in both locations. This was determined through the Monte Carlo ensemble method, which was used to identify significant betas that were processed through the Bayesian confidence interval and the Benjamini-Hochberg method. And this method was developed by my mentor, Isaac Torres. And so once I found the significant QTLs that matched between the 2 locations, I looked through them to find genes that I thought would be important to sorghum. And I specifically focused on heat and drought ones more. This is because both Georgia and Arizona are in high heat conditions, and then Arizona has drought, as we will see in the pictures in a little bit, and also anything that increases water uptake is generally good for plants to begin with. So this is a picture of our Georgia field. This was our small field, not the exact data we used, but the plants look about this tall. I wanted to include this for reference so you can see that they get to about 12 to 13 feet tall on average and as tall as 18 feet. In stark contrast to the plants in Arizona, where they are all basically underfoot, and you can see from the soil that there's very, very dry conditions. So drought is definitely a factor here. And also it's important to note that although these plants are so different in terms of how they've grown, they're actually the same genetic accession. So the difference in this is from environment rather than genetics. And here are my results for the genes that I found to be most notable. Starting with frataxin. Frataxin, its expression is upregulated during drought and salt conditions, and it is also thought to respond to oxidative damage as well as phosphorylation. And dehydrin, overexpression of it can increase tolerance to drought, high and low temperature, high salinity, and heavy metals. It participates in stabilizing membranes, enzymes, nucleotides, and cells under abiotic stresses, some of the stresses mentioned above. And the last one is an inorganic phosphate transporter. I could not find much on it when I looked it up, so that's why there's no description, but I thought it could have some kind of unknown association that we don't know outside of other phosphate transporters. And then here is what I was talking about with the overexpression of frataxin. So you can see this is control and under salt conditions and then under drought conditions as well. And especially in the leaves and especially in drought, there's a large increase in expression, almost double in this case. So it is very clearly responding to drought, and as I'll explain more, it helps the plant. So it is thought to play roles in iron metabolism of both the iron sulfur cluster and heme synthesis, and plants that are deficient in frataxin have been found to have slower growth, increased reactive oxygen species, and oxidative stress markers. And recent studies have found that frataxin plays a role not just in mitochondria, but also chloroplasts. I included images of both on the side. So before, frataxin was only really studied in humans for the most part. There's little research done in plants, and this is because humans get a very debilitating disorder when they have a problem with their frataxin. But as you can see, frataxin has been studied, so they know the regulatory pathway is much better than they do in chloroplasts because it has not been studied in plants much. And then as for dehydrin, it can prevent water loss during stress by stabilizing the cell structures and organelles. There's little description of the stress-induced regulatory network of them, but I included a diagram of an attempt at trying to quantify what it is from the study I cited. And they also combine with metal ions to reduce DNA and protein damage caused by reactive oxygen species, but there's little known about the regulation of their genes. As for further study, there's been no published studies analyzing relationships between AMF and frataxin or sorghum and frataxin, and this is the same for both with dehydrins. I think this could be very beneficial, especially with frataxin, because I thought it was interesting that frataxin was marked to be significant for Georgia. I expected it for Arizona, like once I found out what it does, because it's for drought and salt conditions, but we don't have drought in Georgia or high salinity. So I think there's more important to it outside of the drought and salinity, and anything that increases water uptake is good for plants because farmers will not have to water as much. And I would like to compare the heights of plants with this conserved frataxin gene to those that don't in both Georgia and Arizona. This can be done with dehydrin as well. And I believe it also may be beneficial to study the stress-induced regulatory networks of dehydrins, because as I mentioned before, there's information missing, and it would help us learn more about the plants and perhaps association with AMF. And these are my works cited.
Wonderful talk, and it raises a lot of interesting questions. So one of the things that'll be interesting to do is the Arizona and the Georgia study, had these different fertilizer treatments. So it'll be interesting to look at what genes get pulled out when you take account of the block effects for the different fertilizer treatments. But so that's probably something on your plate in the future.
Yes.
I'd be glad. He'd be glad to take any questions. You can just go to the bottom of your screen and put something into chat, and I will ask Jake about that. So so you found both of these, and I'll just ask my question while you type. You found these sets of genes at both localities then, both at the Georgia locality and at the Arizona, which is, but that's, I just want to verify that's what actually you found.
Yes, I found them at both the exact same SOBIC number for both.
Okay, okay, that's really, because that's sort of a validation in large scale GWAS studies. Usually what they try to do is pick two different sites, and hopefully whatever thing you're looking at shows up in both sites. Do you have any questions yet? No? Okay, I'll ask one more question. Do you have any thoughts, or are you planning to look at sort of allelic effects of these three genes that you found? SNP effects. Is that something you plan to do?
I haven't thought as much about that. I was thinking more about the looking at the graphs for the height data to see if I could find that.
Right.
But I think that is something that I could definitely look into after that.
Right. Okay. So I'm going to stop right now. We're on time. And I want to thank all the speakers today and the support of DOE and NSF. And we will be back here at 1 o'clock for our speakers. And thank you so much. Well, welcome to the 2023 REU Virtual Symposium, and thank you for coming back. This symposium and the work that went into it is made possible by National Science Foundation and the Department of Energy, as well as the support by Franklin College, the Department of Genetics, the Department of Biochemistry and Molecular Biology, and the Institute of Bioinformatics. We have an exciting set of remaining speakers. And the first one is Isabella Wilson. She is also part of the AMF sorghum bioenergy project headed up by Jeff Bennetson. And she's going to talk to us about the relationship between the fungal disease burden of the host plants and the degree of colonization by the arbuscular mycorrhizal fungi. So please start, Isabella. Oh, and let me say a few more things. I should have said that she's from the University of Georgia, and I guess I'm her mentor along with Ms. Shufan Zhang. Okay, thank you. Oh, and please put any questions in the bottom if you when she finishes her presentation. Okay. Okay, go ahead, Isabella.
Okay. Hi, everyone. I'm Isabella. Like Dr. Arnold said, I'm talking about the relationship between disease burden and arbuscular mycorrhizal fungi in sorghum bicolor. Okay, so arbuscular mycorrhizal fungi are small biotrophic fungi that form symbiotic relationships with plants by colonizing the plant roots. And the fungi extend a branch network called hyphae, which infiltrate the cortex of the root, which is the layer of cells surrounding the innermost portion of the root. And the functional unit of this relationship is the arbuscules of the AMF, which are round structures that AMF form inside of the cell wall, the outside of the plasma membrane. And this is where the fungi facilitate the uptake of nutrients for the plant, and the plant in return supplies the fungi with carbon that it needs to survive. And one example of a plant that AMF colonizes is sorghum. So sorghum is a gluten-free, drought-resistant cereal crop, and Dr. Arnold and his collaborators are currently investigating the relationship between AMF colonization and sorghum. And part of this project includes identifying regions of the sorghum genome that affect this relationship, as well as other plant phenotypes, including biomass and disease resistance. Regions in Africa and Southeast Asia rely heavily on sorghum, so understanding how to improve sorghum health and yield is very important. However, disease is very common in these plants, and it dramatically affects crop yield, which can pose a great issue for these regions. So going off of that, the research goal for this summer was to quantify AMF colonization in root samples and then relate this back to disease burden, which is thought to be reduced by the presence of AMF. And along with this, I'd like to propose a model to explain the relationship between AMF colonization, disease burden, and sorghum genotype. So based on previous literature, I hypothesized that plants with high disease burden will have less colonization compared to plants with low disease burden because the presence of AMF has been shown to increase disease resistance based on the idea that AMF improves the overall health of the plant. So for this project, the disease that I focused on was target leaf spot, which is a necrotrophic fungal pathogen that manifests in red and brown spots on the leaves of the plant. And so target leaf spot was a really big issue in our sorghum field from last summer, so that's why I decided to focus on this for my project. And it is known that disease susceptibility in plants is affected by the plant's genotype, but not much is known about target leaf spot resistance genes because the pathogen is not well understood at the molecular and genetic level, and there's not a reference genome for it. Okay, so like I mentioned, for this project, I used data from our sorghum field from last summer, where we had 86 sorghum accessions, which is another word for a set genotype, planted in a randomized 3-block design with 6 plants of each genotype in each block. And 1 to 2 plants from each accession in each block were harvested and scored for a variety of phenotypes or traits, including disease. So this is a heat map basically showing the spatial distribution of disease severity across the field, and disease incidence is pretty randomly scattered. There's no pockets of super high disease or super low disease. So that's just showing that there's no obvious environmental effects that are causing one area of the field to have a much larger disease burden than others. Okay, so then here's a distribution of the disease scores across all the plants that were sampled, and the target leaf spot distribution does follow a relatively normal distribution, which is good. And then the second graph is showing the disease distributions between blocks, and they're literally the exact same, which is great because that means that, again, there's no large environmental effect that's causing one block to have more disease than another. And then this is another box and whisker plot that's showing the distribution of disease for each specific genotype. And the reason I'm showing this is just because it points out that some accessions have consistently low disease or consistently high disease, and this suggests that there is a genetic basis for disease burden, which is a point that I'm basing this project off of. Okay, so for my data collection, I chose 10 accessions that had consistently high disease and 10 that had consistently low disease. And for all of these, I got the root samples that we harvested last year and used Chuffon's procedure to clean, clear, stain, and image these samples with our high throughput microscope at 10 times magnification. And this is just an example of what one of these images looks like. And I used an intersection-based method based on the classic McGonigal method to calculate a percent colonization score. And so I created a standardized representational eyepiece at 200 times magnification, which is the magnification used in the McGonigal method. And I did this by adopting a standardized grid set to the pixel length of 20%, the length of a large image file, and then creating a circle that extended to the border of one of these squares in the grid. And then the size of this circle remained constant for each image. And then the circle was passed vertically down the root, and whether or not a crosshair perpendicular to the surface of the root, which is the red crosshair here, passed through a region containing any AMF arbuscules was recorded, and the surface, or each pass contained an independent segment of root. And I didn't include any passes that went through root that didn't have any cortex, which you can see the region like right to the right of that circle is very thin, that doesn't have any cortex. And so I didn't count any of those passes because cortex is a prerequisite for the possibility of colonization. So this is just that same image, a little bit larger, and this is from a high diseased root sample. And then this is just a very zoomed in version of that photo where you can see some of the AMF structures. All the like dark blue box brick type structures are arbuscules, which is where that nutrient exchange is occurring. And then the little strings that seem to be wrapped around the root are the hyphae, which is the initial step in colonization. And then this is another portion of that same root image that just has very defined hyphae, and it also has these very defined circular structures that are vesicles. And that's another indication of AMF colonization because the vesicles are formed when the plant cells have extra nutrients and the vesicles hold the extra nutrients. And that would only occur if there were the AMF like supplying these extra nutrients to begin with. And then just for reference, this is an example of a low diseased root that had virtually no AMF in it. And you can see the cortex is much clearer. Okay, so this is a distribution of the percent colonization for the 60 samples that I measured, and it is right skewed, but that does make a little bit more sense when we look into the data a bit further. And then here is another box and whisker plot that is plotting the colonization of each individual genotype, and there is some variation in the amount of within group variation, but when I plotted the variance, it did follow a normal distribution, which is good. And then this is the really interesting part. So I did a t-test to compare the percent colonization between the high disease and low disease groups, and there was a very significant difference. But the difference is the other way than what I was expecting. I was expecting the high disease plants to have less AMF, but they actually had more. So this kind of changed the way I started thinking about my project. And then I regressed these two variables against each other and found that the percent colonization can explain 17.29% of the variation in disease burden. And this was a significant relationship, but it's a pretty weak correlation. So I started thinking a little bit deeper about this. And so I went back to my original hypothesis and asked, even though the relationship was reversed, is it still mediated by genotype? And to test this, I did a mediation analysis. And with this, you'd expect that the combined effect of colonization and genotype would have a larger correlation than either of these two variables alone. But you can see that genotype alone had a little bit of a higher correlation than the combined variables. And although this wasn't a significant difference, you would want colonization and genotype to be significantly higher than genotype alone. So I had to rethink my model again. So I came up with the idea that the difference in disease burden could be due to genetic differences in the plant immune response. And so plants detect pathogens through compounds called microbe-associated molecular patterns. And when AMF initially colonizes, it does activate an MAMP, which is molecular or microbe-associated molecular pattern. So MAMP triggered immune response, but the cells rapidly downregulate this response when they realize that the AMF is symbiotic. And research indicates that AMF and target leaf spot activate the same immune pathway, which is the jasmonic acid pathway. And this suggests that one genetic variant could have similar effects on each of these fungi's ability to infect the plant, which could explain why plants with high AMF also have high target leaf spot. So I wanted to see if there were any genetic variants associated with AMF colonization that could also explain the difference in disease burden. And so to do this, I did a genome-wide association study, or GWAS, which fragments the sorghum genome into chromosomal regions and then tests if the variations in these segments between genotypes can explain the variation in AMF colonization. And so to do this, I used a program developed by Isaac Torres, and it identified 120 regions that were associated. And there's many genes in each of these regions, and any variant in any gene could be the one causing this association. So I basically just had to comb through this list of genes and pull out ones that seemed like they might have an effect on disease. And I do want to acknowledge that this was a pretty rough analysis, just given the thousands of genes in the output and due to the lack of time to actually go through and properly research every single one of these genes. But I did pull out 4 that seemed to be very important. Okay, so the first one is a disease resistance responsive family protein. So this protein is known to couple plant metabolites called phenols into lignans, which are bioactive compounds that serve as a physical barrier to pathogen infection by thickening the plant cell wall. And phenols are synthesized in response to stress, and an example of stress could be disease. And this gene is highly conserved among vascular plants. And the next one is a jasmonate Zym domain containing protein, which is a signaling molecule in the jasmonic acid pathway. And it forms a complex with a JA responsive transcription factor and serves as the first signaling molecule in the cascade. The next 2 are related to chitin, and chitin is a critical component of the cell wall of fungi and also serves as an MAMP. And a major function of chitinase is to degrade the cell walls of invading pathogens, so it makes sense that a variant in this gene would affect the affection rate of both AMF and target leaf spot. The last one is a chitin elicitor binding protein that recognizes chitin and binds to it and then transmits this signal to the rest of the cell to let the cell know that there is a pathogen present. Okay, so to conclude, AMF colonization can explain 17% of the variation in target leaf spot. And the hypothesis that AMF colonization and disease burden are correlated because genetic variants affect their ability to infect in similar ways is supported by the MAMP and JA genes that showed up in the GWAS. And ideally, I would like to replicate this experiment with a much larger data set to see if this trend still holds true. And I'd also like to run a GWAS for disease burden instead of just AMF colonization, and then compare the polled genomic regions and see if there's any overlap. And I did initially try to do this, but it didn't work properly due to the nature of my data set because my disease data was very highly skewed on either side. So if I were to do this and the trend still holds true, then I would like to do a greenhouse experiment to experimentally test this relationship. But these findings are really important because it suggests that plants that colonize AMF better might also be more susceptible to disease, which suggests that the ideal genotype for agriculture and regions that are highly susceptible to disease might not be the genotypes that colonize AMF at the highest rate. And this is also the first time that this high disease, high AMF trend has been observed. So I think it's a really important area of future research to look into. Okay, and then here are my references.
Much. That was wonderful. So also to test your model, have you thought about any using any knockouts in sorghum as
Yeah, I think that would be a really great direction to pursue in the future once this like relationship has been validated in a larger data set, just to see if these genes that I'm pulling are actually the ones associated. I think it would also be really cool to go through and run regressions for every single variant in these genes to see if we can find the one SNP or the one indel that's causing this relationship. That just takes a very long time to test every single one of those, and was kind of outside the scope of this summer. But that would be really cool to test in the future.
I have a question for you here. And people, you can put in the questions. One of you has already done so. Great work, Isabella. One question: what is the difference between, it's biotrophic and necrotrophic fungi? This is from
So biotrophic fungi, so AMF is an example of that, and it requires that the host plant still be alive to supply the fungi with the nutrients that it's trying to get out of the plant. But a necrotrophic fungi like target leaf spot actually kills the plant in the process of getting like the nutrients that it's looking for. So that's why necrotrophic fungi like target leaf spot are so detrimental to the plant health, because it is killing the plant in the process and reducing the like biomass and the yield of the plant.
Okay, great. And again, you can, I'll see questions pop up. The other, I think, oh no, that's the one I already just read. Another question I had is on your scoring system that Bushar helped us with. Was the scoring system you used for fungal disease? It was fungal disease. Was that aggregated across four or five, or just the target leaf spot?
I just used target leaf spot scores, but in the data collection from last year, we have scores on 5 different diseases, but target leaf spot was in almost every single plant that we sampled, and the others were like much more scarce. So that's why I focused just on the target leaf spot.
Okay. But there is the potential there, at least, to go after, sort of a kind of replication where you could look at another pathogen and see what it's doing. So that's great. And okay, and I think I guess I'm 3 minutes over, and we're getting into Michael's time. So I will stop there, and thank you again very much for a wonderful talk.
Thank you.
Okay, it's my pleasure to introduce Michael Douglas. He is at the University of Georgia. I believe he's a sophomore coming up to be a junior this fall, right? Yep. And his mentor is Mr. Isaac Torres, and he's going to be looking at how we model the era in a GWAS study using environmental variables. And it's my pleasure to turn it over to him, and he'll lead us in this investigation.
All right, well, thank you for the introduction, Dr. Arnold. So my project is titled Modeling the Environmental Component and Genetic Variation in Height in Sorghum with Weather Data. And some background is sorghum bicolor is a grass crop used for food, feed, sugar, and biofuels, and it's also a staple crop for millions of people in sub-Saharan Africa and South Asia. And these areas are highly susceptible to droughts and high temperatures, which can cause significant yield and loss to the crops. And to achieve food security in these regions, drought and high temperature stress tolerance should be genetically improved. So previous studies have shown that increases in diurnal temperature changes can result in shorter plants especially between 40 and 53 days after planting. And diurnal temperature ranges are actually the temperature difference between the minimum and the maximum temperature in a 24-hour period, so within a day. And knowing how environmental conditions affect sorghum growth and height can complement the AMF and sorghum GWAS study as a whole. So the overall goal of the AMF sorghum project is to try to find the genes that determine the amount of AMF colonization in sorghum roots under many different environmental conditions. And my project aims to analyze the genetic component and height variation while also accounting for weather conditions that affect the development of sorghum, such as rainfall and maximum daily temperature. And my specific GWAS involved taking height data from our field in Maricopa, Arizona, and running it through ensemble methods in order to find significant chromosomal regions affecting height. So my hypothesis is that including environmental data in the GWAS analysis could be better for finding significant genes that pertain to sorghum height, development, and stress tolerance. And that's just a picture of me in our field here in Georgia. So some materials from Campbell Scientific, I needed a CR6 data logger, a weather station, and a CS650 soil moisture and temperature sensors, which is that thing with the sticks, and height data from the 2023 Arizona field, weather data from the Maricopa Agricultural Center at the University of Arizona, and the GWAS analysis program developed by my mentor, Isaac Torres. So methods. So last year, weather data was also collected by another REU participant. Unfortunately, the battery for the weather station died and the data was lost due to data being collected too often. I think it was every 30 seconds. So this year I programmed the CR6 data logger to collect data every hour and take the average and standard deviation of the data every 6 hours and save it. So data was collected on air temperature, soil moisture, electrical conductivity, permittivity, and soil volumetric water content. And I conducted a sensor calibration experiment with all 4 sensors placed in a tub of water just to make sure nothing wacky was going on with the data. And each sensor was buried about a foot deep with a sensor in block A, 2 in block B, and 1 in block C. So the picture on the left, you can see the sensor calibration test with all 4 of the working sensors in a tub of water just to make sure none of them had anything weird going on with their data. In the middle was the weather station after we set it up in the field. And on the right is a sensor buried in the soil about a foot deep, like in between 2 rows of plants. So here, there's a map of our field here in Georgia, and circled is where we buried the sensors and where the weather station is located. As you can see, there's a sensor in block A, 2 in block B, and 1 in block C. And I tried to evenly space them out to get a good sense of the soil conditions of the field, and the weather station is just off to the side of block C. So my experimental design, I built a linear regression model for height with maximum daily temperature and cumulative rainfall for each accession. As you can see, the 2 models, the only difference between the new model and the original is 2 new coefficients that represent rainfall and max daily temperature. And I fit both these models using ensemble methods and compared them. And the Monte Carlo ensemble method was used to identify regression coefficients that were then processed through the Bayesian confidence interval and Benjamin-Hochberg methods. And this gave me significant chromosomal regions, which were then evaluated to find significant genes that affect sorghum height and development. So some controls were the replication of accessions, blocks, localities, weather data, and calibration of sensors. Some preliminary data that I needed was height data from around 3,600 plants from the Arizona field, and 2023 temperature and rain data from the weather station at Maricopa Agricultural Center. And that picture is from the field out there in Arizona. As you can see, it's a desert. So yeah, this is the weather data from Maricopa, Arizona. As you can see, it got to over 100 a lot of the days. I was actually in Arizona from June 16th to June 22nd, and the graph does not lie. It was over 100 like every day I was there. It did not rain one time the entire time I was there. As you can see in the graph on the right, it rained actually one time the entire 2 months from when the plants were planted to when we took the height data. So a cumulative total of 0.07 inches, which is nothing. So this is results. This is probably the most interesting gene I found. I found a dwarf gene, and plant height is traditionally reported to depend upon the action of 4 independent dwarf genes. DW1 to DW4, having partial dominance for tallness and additive effects. And I found this gene in bin 2453 on chromosome 9, and there's the gene. And so this is DW1, and it encodes for a putative membrane protein involved in the regulation of cell proliferation in the internodes. And cell proliferation is basically an increase in the number of cells, which leads to growth and relates to height. So since the gene has additive effects on height, it's interesting to see that only DW1 showed up while DW2 through DW4 did not. And this gene only showed up in the second GWAS analysis, which accounted for rainfall and temperature. So that's interesting. And although DW1 through DW4 explain most of the observed phenotypic variability for sorghum plant height, the existence of a fifth dwarf mutant gene, DW5, was observed in 2019. And I actually read that when DW5 is expressed, it actually causes plants to usually be shorter. So that's cool. And two more genes that I found a MADBOX protein. And MADBOX genes are a key family regulating many aspects of plant growth and development, such as flower development, substance and energy metabolism, signal transmission and transduction, and dormancy processes for plants. And I found this gene in bin 108 on chromosome 1. There's the gene, and this gene was found both and in the first and the second GWAS analysis. So it is pretty significant in relation to plant growth and height, I believe. And the second one I found was a transcription factor, and they play a crucial role in plants' adaptation to stresses because they act as master switches and regulate the expression of stress response genes. And I found it on bin 463 on chromosome 2. And this gene is actually a part of the AP2 subfamily, which is amongst the transcription factors that respond to abiotic stress, and expression causes drought tolerance in many species. So this gene was downregulated in drought-tolerant cultivars in response to drought and was found only in the first GWAS analysis, which did not include weather data. So for further study, unfortunately, I was not able to do a GWAS analysis on the high data from our field in Georgia this year, but I did, however, collect all the soil data and weather data needed for future analysis. And I can continue this project by running 2 more GWASs with and without the weather data on the high data in Georgia, and I can then compare them to the 2 GWAS analysis I ran on the high data from Arizona. And a potential future hypothesis is that sorghum crops in cooler, humid environments with moderate amounts of rainfall will grow taller than crops in hot and dry environments, and higher rates of rainfall in Georgia will lead to slightly cooler temperatures, not too much cooler, which will promote growth in the crops because of decreased heat stress. And the environmental effects and variability in height will be larger in Georgia than in Arizona due to the climate stability of the desert. As y'all saw, it really did not rain out there. So yeah, that's all I got.
That's wonderful. Thank you for that wonderful presentation. So again, you can put questions at the bottom of your screen. I'll just interject one of mine. So one of the things about the Arizona data is that we had 12 blocks out there, with different fertilizer treatments. So do you plan to work with Isaac on maybe including block effects into your analysis?
Yeah, I think that would be really important and cool to see how the levels of nitrogen and phosphorus treatment would affect the height. And
right.
Then I could compare it to the Georgia field from last year, which also had the phosphorus and nitrogen treatment. So yeah, that could be a future project.
I think that'll also be interesting because we can go back and look at if that removing that effect has an effect on picking up the other dwarfism alleles. We might have lost them because of the variation between blocks. So yeah, I think that would be great. Let's see. I looking here, and so the and you did mention that some of the genes you looked at or you pulled up in Arizona, you were able to, I guess, validate with Jake. They actually showed up in the Georgia analysis. Was okay. So that's also interesting. That's another kind of independent validation as well. Okay.
I think Jake actually had more overlap between
Right.
the Arizona and Georgia field than I did between the
Right.
run without the weather data and with the weather data. So
Okay.
yeah, cool to
That's
see.
great. Okay, I think we'll stop there and move on to our next speaker. And this is another person who is working on the AMF sorghum project. And her name is Anna Satori, and she's from the University of Georgia, and her mentor is Isaac Torres. And she's going to talk to us about what the roots look like in sorghum. So I'll turn it over to her.
Yes. Thank you, Dr. Arnold. Yes, my project is on the root morphology of sorghum bicolor across different accessions. A little bit of background. I'm a part of, as Dr. Arnold said, his lab, which is doing a multi-year, multi-PI experiment studying the relationship between sorghum bicolor and arbuscular mycorrhizal fungi. You can see that the sorghum bicolor is depicted on the right. It is a gluten-free cereal crop that is relatively drought tolerant, and we're interested in it because it can produce biofuels or biobutanol as a resource used for energy. We are looking for the relationship between this plant and the arbuscular mycorrhizal fungi, which is the fungi that form an endosymbiotic relationship with plants. Actually, 80% of all land plants have this relationship. It's very essential to the plant as it's a give and take relationship where the fungi provide the plant phosphorus and nitrogen, and in return, the plant provide the fungi carbon for energy. The AMF have also been seen to improve the drought tolerance and the tolerance to disease in plants, and also aids in the soil structure a lot, as well as carbon sequestration, which is all very important in developing more sustainable agriculture. So our lab is interested in finding the genes that are involved in this relationship and see how we can use this relationship to optimize agriculture in the future. Here's a imaged root of sorghum, one of the fine roots. You can see in blue, this root has been stained with blue, and the blue is actually the AMF. So there's a lot of fungi in this specific sample. It's very highly colonized. You'll see on the left, the circled structure is actually an arbuscule, which is where the fungi gets its name from. This is where the important nutrient exchange takes place. A little bit about my own project, as this is a very important system, as all economically important crops form this relationship. It's important to understand it and, again, see how we can optimize it. I'm interested in the root morphology, the actual physical features of the roots, to see how we can see which root structures are best for the AMF colonization, so that we can find and possibly select for these root structures later on as a way of selecting the phenotypes of underground traits to depend and rely on these AMF for nutrients instead of us providing fertilizers and waters and a lot of input. So if we can figure out how to use these AMF, again, we can develop a lot of less input agriculture. It has been shown that there are some influencing factors in the environment on this root morphology. It's actually been shown that bacteria can cause more lateral branching in the roots, and AMF have even been shown to induce less fine root hairs. So we know that there is an environmental effect on these morphological structures, but we don't know exactly how much of these root morphology is derived from the genotype of the plant rather than the environment. So that's like the first step in my experiment, is to, because my overall questions is, does plant genotype affect root morphology? And so that we can then figure out, does root morphology affect AMF colonization? So we can then optimize it. I have derived 3 hypotheses from these overarching questions. The first being root morphological traits will be more similar between genotypes than within genotypes. I performed it in ANOVA to do this, which I'll be talking about later. My second is that root morphology and plant fitness are related. Some traits are correlated with better fitness. And my third is that some root morphological traits are better suited for this AMF colonization than others. To begin to do this, I needed to quantify all of the root morphological traits. I did this using a software called Digital Image of Root Traits, DIRT for short, ironic, which is a high throughput computing collaboration platform for phenotyping the roots, created by Alexander Bush, and I was advised by Isaac Torres, a PhD student, on how to do this. I have selected a few traits that I was most interested in studying, listed here. I went through a lot of root images, hundreds, and they all look similar to this from the 2021 field harvest. And I had to select a much smaller sample from these. I ended up choosing 32 different accessions from the BAP or Bioenergy Association panel with multiple plant repeats in each of the accessions. I ran the images through DIRT, and what you're seeing below is what images go in. It would be the original, and then DIRT gives me the second two images. The first, the black and white middle one, is the one that the most of the measurements that I received were derived from, and the second, the root skeleton, is where I got my depth trait from. I had to dip my toe into a lot of different software programs to do this, including Sapelo, MATLAB, Python, and R, and this was a large part of my summer of figuring out how to do this statistical analysis on these programs. To understand this nurture versus nature question of how much of the root traits are determined by the genotype versus the environment, I needed to do an ANOVA test, which is a one-way analysis of variance, basically showing the significant difference of the means between the different groups or the different genotypes. Here, or PIs, you'll see later. I needed to fit some assumptions before I did the ANOVA test, which, for example, the normal distribution, all of my data had to be normally distributed. I did a QQ plot, which is what you're seeing on the right, the probability plot for each of my traits, and received the top graph shows an ideal result, showing that it has normal probability, and I'm able to use this trait. The one below it, though, the average density, showed tailedness and kurtosis. As you can see, it didn't fit on the line, and I was not able to use average density because it did not have normal distribution. I also analyzed for homogeneity of variance, which I just looked at the box plots, and I also used the box plots to pick out outliers that I needed to have removed. The image on the left just shows that I had 31 traits to choose from, and they all had a variety of distributions that most of which I was not able to use. All these traits are independent, as one root trait doesn't really determine another root trait. My results with the 5 traits that I was able to analyze, as I couldn't use average density, I received results of the ANOVA. This one is an example of a significant result, meaning it rejected the null hypothesis that there was no significant difference between the genotypes versus within the genotypes. In this graph, you'll see that though this box and whisker plot shows that for each of the PI or the accessions, they show a lot of variation either in between the genotypes as well as with the longer boxes within the genotypes. I calculated the broad sense heritability, which is within group variance divided by total variance, and got 0.6 for this one, meaning that it's 60% heritable, which is interesting. I did this for the other 4, and it was intriguing that 2 of them did not pass or did not show significant results from the ANOVA test, meaning that there was not significant difference between the genotypes rather than within the genotypes. Meaning that these 2 traits, median root tip diameter and median width of the root system, are possibly more affected and influenced by environmental factors, possibly AMF itself, than the genotype of the plant. Projected root area and root depth skeleton, however, did pass these, showed significant p-values, meaning that these are determined more so by genotype than by environment, with broad sense heritability of around 0.7 for each, or 70% heritable. I then moved on to my second hypothesis of seeing how the correlation between the traits and the fitness looked, and to no surprise, they all showed positive correlation with canopy dry weight, which was my measurement of fitness, as it is just the above ground mass of the plant dried and weighed, showing basic success of how the plant did. So it shows that the more root tips, or the larger the plant, the more root tips that it has, which makes sense. So the R² value of 0.1. I also did this with projected root area. This one showed a slightly more significant R² value of a 0.2, showing that it was more positively correlated. I also did this with root skeleton depth, also positively correlated with depth with weight of the plant. And I did median root tip diameter, even though it didn't pass the ANOVA test. It's an intriguing trait to me, as previous literature have noted it as a important morphological trait for AMF, as the thickness of the plant correlates with the amount of cortexes in the plant, which is where the cortex is where the AMF symbiosis takes place. I did meet a lot of limitations throughout this experiment. For example, the first step of the experiment was taking the images of the plants, and this was done in very harsh conditions, as it was in the field right after we dug them out of the ground, and there was a lack of standardization with how the images were taken, and this possibly could have skewed the results that dirt gave me from the quantified root traits, as dirt requires very specific, very standardized images. I think that a simple fix of this is to redo this experiment and develop a protocol of how to do this in a better way. I was also limited in the number of plants that I had per succession, and I think that in another experiment I could create a larger group within each succession, so that the data will be more significant in determining how much of the percentage is determined by genotype or environment, as they'd have more to compare with. I also didn't even get to my third hypothesis, which was the AMF colonization, which is the next step in the process. I am just at the very beginning of this larger experimental idea, where once we know that these morphological traits are determined by genotype, we can then isolate and find which of these traits are most beneficial for the AMF, which then we can find using a GWAS study, which I plan on doing, which traits correlate or associated with which genes, so that we can understand more of the genomic process of developing these morphological traits, which we can then use to either select a breed or even use transgenic experiments to create plants with the optimal root traits for this AMF relationship, so that we can, I guess, create more sustainable agriculture and have less input farming. I would like to acknowledge my mentors, Isaac Torres and Jonathan Arnold, as well as all of my other fellow REU students. And these are my sources.
Thank you very much. Again, people, you can put your questions in the chat at the bottom, and please do. While you're doing that, I'll just horn in and ask a few questions. So, you had to pick your way through the samples last year. Do you have any, what sort of initial things would you like to do to improve or try out to improve the imaging for this coming fall?
I'm excited to develop this protocol this upcoming field harvest in September. I think that the most important aspect of it is the lighting of it, as it illuminates the roots in different ways. And last year we took it with a phone that automized the lighting and the focus, so each plant showed slightly different images, quality, and lighting.
Right.
I think that if we were to standardize all of this with a possibly professional camera and lighting that we fix in, as well as as a neater background, as there was like dirt chunks in the last images, I think that the dirt quantified results would be a lot more accurate.
Okay, let's make sure we order the professional camera now.
I'm going to do my research on it. We'll see.
Okay. And so, and then you're presumably also going to take samples from the small field GWAS and try to look at hypothesis 3? That's your
Yes.
your
I still think that's
goal.
possible to do with this experiment. I
Right.
didn't have enough time to finish that,
Right.
but I
Right.
think seeing the relationship between high colonization and specific root traits will be very beneficial for the next experiment that
Yeah.
comes.
And those samples exist, right, from
Mhm.
last year.
Yes,
So
sir.
they're there, and I think they've even been processed on the high throughput microscope, robotic
They have not
microscope.
yet.
Oh, they
I would have
haven't
to
been
do that.
processed.
Yeah.
Okay,
I
so
need to
you'll have as a
clear and
priority
stain them and
to
image
do
them.
that.
Mhm.
Okay. Okay. So I'm just looking for questions here. I think we're right at the right time for the last speaker. And the last speaker is going to be a virtual presenter on a virtual meeting. And I'll introduce her, and then the virtual presenter will present. So, the last speaker is Daniel Watson. She is from Clark Atlanta University. She was mentored by Dr. Annie Chung, who is on the AMF Sorghum Project. And she is going to be looking at you might say, the community ecology of AMF with other kinds of fungi. And I'm just going to let her start talking. Okay, thank you.
Good evening, everyone. My name is Danielle Watson, a rising senior at Clark Atlanta University, and this summer I was studying ecology of fungal disease and insect herbivory in a Piedmont prairie through the Dragnet Global Project through Chung's Plant and Microbioecology Lab. What is Dragnet? Dragnet is the disturbance and resources across global grasslands experience, which is aimed at assessing the site-specific factors that influence disturbance recovery and community assembly in herbivorous-dominated ecosystems. And this summer, I collected data to assess plant disease and insect damage in the local Athens Dragnet site. What is the Dragnet experience? It is comprised of 5 separate treatments spanning over 25 different plots, varying from control, disturbance, NPK, which is basically just nutrients, NPK and disturbance, which is nutrients and the plot itself being pulled up and disturbed, and then NPK plus the station, which does not actually happen for, I want to say, another 1 to 2 years. So as of right now, that is just another NPK plot. And then some hypotheses that I had or research questions was, will the addition of the nutrients affect the amount of plant disease, and will disturbance affect the amount of plant disease? So what was I studying? And what I was studying was called Panicum oleosanthus. And that is basically a narrow-leafed grass that is infected with the black rust that I was looking for, which is the fungus that I was looking for. And I use that as an indicator to understand if the treatments of nutrients and disturbance actually were effective. And what does this disease look like? It looks like the image in the corner left. And basically, I surveyed and collected data from all 25 plots to see what of the plots and treatments had this present disease. Now, here, my data collection method, I was to identify the panicum, which is the bunches of the grass to the left of my sneaker. I would actually close my eyes and select the plant. You have to basically, in order to effectively locate the disease without being biased, I had to close my eyes and kind of pick. It's kind of funny, but it was the most effective method. From there, I would select the level of disease. So it ranged from 0 to 100. And how I was able to select was if I were to spread out the apparent disease on the entire leaf, what range would it cover? And lastly, I would just move on to the next plot after I've collected 40 leaves. And my results show that within NPK cessation, NPK treatments, and NPK disturbance, the plant disease was lower, and in control, as obvious, it was higher. What are the next steps? So some possible next steps is to further progress my research if I'm ever so fortunate to come back to the program next year. If not, another student who decides that they want to further their knowledge or just any information about the Panicum plant, they would just follow that along with any of their articles or sightings. In addition, I would love to keep in contact with Annie and everyone working with the plant and just see the next steps in how things like the NPK cessation will affect it, or further disturbance, or just nutrients on its own. In addition, this summer, I worked on identifying insect damage within another plant, which is actually a forb called crownsbeard. And crownsbeard is an apparent winged stem, making it easy to identify, which is basically this image right here. What did I look for? Similar to the previous plant, I was looking for damage. However, I was looking for insect damage, which allowed for a deeper understanding of how treatments affect the insects and plant enemies in general. And similar to my last research questions, I wanted to know, will the nutrients and disturbance affect the way insects damage the plant? And similar to the last plant, I made sure to identify the crownsbeard, close my eyes and select the level of disease or damage, and move on to the next plot. Now, for crownsbeard, it was a little harder to get up to 40 for a lot of the plants because the plant itself is not it doesn't contain a lot of leaves to work off of. And it actually was not present in a lot of my disturbance plots. So that left out a lot. In addition, it was also not present in the center or in the plot, but it would be around, but outside of the plot. So with that being said, the results basically showed that within NPK in disturbance, it was lower. However, as you can see on the side where it says insect damage, it was not a lot to work off of. So some next steps would be, if I'm ever so fortunate to come back to the program next year, is actually redo this specific forb, because there was not a lot to work off of. So maybe potentially only counting a certain amount of leaves instead of doing 40 for one plot and 25 for another and 15 for another, just to give it an overall better chance of being surveyed. And yes, and then some of my acknowledgments, and I have a lot, is Dr. Jonathan Arnold, Dr. Annie Chung, Dr. Alex Strauss, Dr. Rebecca Abney, Christine Milley, and the National Science Foundation for funding this REU program. Thank you, everybody. I'm so sorry I couldn't be there in person to actually do this, but I enjoyed myself at this program this year, and I, God willing, will be there next year. Bye, guys.
Well, thank you, everyone, for joining us today to see all these exciting research projects by the REU participants. I want to thank, again, the National Science Foundation and the Department of Energy for making all this work possible. I'd also like to thank the support of Dr. Dean Dorsey from the Franklin College, as well as the supporting units, the Department of Genetics, the Department of Biochemistry and Molecular Biology, and the Institute of Bioinformatics. We're very grateful to have had this partnership with these wonderful REU participants, and we were grateful for this exciting summer and the research findings which they provided in their work. And I hope this will provide an inspiration to other undergraduates out there for the opportunities in multidisciplinary biological research. Thank you again, and have a good rest of your summer.